everyone and welcome to Miss Estruch Biology. This video is going to be a big one. It's AQA Paper 2. Everything you need to know for that exam paper. So get settled in, hit ×2 speed if you're in a rush, or use the time codes along the bottom. And if you do need any extra help, don't forget to check out the resources in my description below, such as my A-level notes or my flashcards to really help boost your grade. But for now, best of luck with your revision and let's get into it. So the first thing to do with photosynthesis is knowing where it occurs and just a mini recap on the structure of a chloroplast, because you need to know where each stage of photosynthesis happens. So here on our chloroplast, we can see the thylakoid stacks, and the thylakoid membrane is on the outside of that. And that's where you have these folded membranes where you have all the proteins embedded for the electron carrier chain, and also ATP synthase. And that is where the light-dependent reactions occur. A stack of them is called a granum. The fluid part in the centre is called the stroma, and that's where the light-independent reactions happen. And you have lots of enzymes there for those. And then we have the inner and outer membranes as well, which are controlling what can enter and exit. Photosynthesis is a two-stage reaction. We have the light-dependent and the light-independent reactions. And the light-dependent reactions happen first, and as the name suggests, it requires light. This happens on the thylakoid membrane, or you could call that the granum. In this light-dependent reaction stage, light energy is used and water to create ATP and reduced NADP. And those two products are required for the light-independent stage, which comes second. So there's 4 key steps here. We have photolysis, or sometimes it's pronounced photolysis to remember the stage, photoionization of chlorophyll, and chemiosmosis. Now those are actually the 3 processes. Production of ATP and reduced NADP is the outcome, but AQA states these are the 4 key stages that you need to know. So starting with photolysis or photolysis, photo refers to light, lysis means splitting. So photolysis of water means light energy is absorbed and then that splits the water. And it splits it into oxygen, electrons and protons, which are hydrogen ions. What then happens is the protons are picked up by NADP to form reduced NADP or NADPH, and that is then used in the light-independent reaction. The electrons are passed along a chain of electron carriers, and the oxygen is either used in respiration, but it's not used in photosynthesis. So if it isn't needed for respiration, it will diffuse out through the stomata. We'll go through in a bit more detail what happens to the protons and electrons when we get to chemiosmosis. Photoionization of chlorophyll, again, is to do with light, because we've got photo. So light energy is absorbed by the chlorophyll, and the ionization part is referring to the fact that the light energy results in the electrons gaining energy. And we describe that as them becoming excited and raising up an energy level. And that causes the electrons to leave the chlorophyll, and therefore the chlorophyll has been ionized. Some of the energy from the released electrons is used to make ATP and reduced NADP in chemiosmosis. So let's have a look at chemiosmosis then, because this then links it all together for the light-dependent reactions. So we've said that the electrons gain energy from the chlorophyll, will be released from the chlorophyll. What happens is those electrons move along a series of proteins embedded within the thylakoid membrane. And as those electrons move along the proteins, they release energy. And some of that energy is used to pump those protons from the stroma across a protein and into the thylakoid lumen. This then results in a concentration gradient being built up, but we actually call it an electrochemical gradient because we have a charged molecule, so we have ions there. So now we have this electrochemical gradient that enables the protons to move by facilitated diffusion back down their concentration gradient to the stroma. But the only protein that they can attach to is ATP synthase. And as the protons attach and move through ATP synthase, that enables the enzyme, which the protons move through, to phosphorylate the ADP into ATP. So that's how the ATP is produced. We then have the protons back on the stromal side, and this is how we create NADP that is reduced, or reduced NADP. So the NADP coenzyme picks up the electrons from the end of the electron transport chain. It picks up the protons after they've passed through ATP synthase, and that reduces NADP, which makes it NADPH. So now we've created ATP and reduced NADP. Those two molecules are used in the light-independent reactions, also known as the Calvin cycle. This occurs in the stroma. It requires enzymes, and the enzyme Ribisco is the enzyme that you need to know. And because of that, this stage is temperature sensitive. But it doesn't require light energy for the Calvin cycle to occur. Now, the Calvin cycle uses carbon dioxide as well as the two products from the light-dependent reactions, and we create a hexose sugar. The ATP that is needed is hydrolyzed to provide energy for the reaction, and the reduced NADP is used to donate the hydrogen to reduce the molecule GP within the cycle. So let's go through this cycle then. The first part of the cycle we'll look at is how the carbon dioxide enters the cycle. So carbon dioxide reacts with RuBP, and that is a 5-carbon compound. So all of these yellow circles are representing carbon molecules. So we can see we've got a 5-carbon compound reacting with carbon dioxide, which only has 1 carbon. That would result in a 6-carbon compound, but actually it's unstable, so it splits into 2 3-carbon compounds, and that's our GP. So we have 2 lots of GP. This is then where one of the ATP molecules is used, and it's where the reduced NADP is used. GP picks up the hydrogen, and therefore NADP is reformed, and the GP is now reduced to form TP. That reduction requires energy from ATP. The TP is then used for 2 things. One of the carbons is removed from these 2 times 3 carbon compounds, and it goes towards creating a hexose sugar. That will leave behind 5 carbons, but as a 3-carbon compound and a 2-carbon compound. So ATP is needed to form those back together, or in other words, regenerate RuBP. Now, that is just one round of the cycle, and that only created 1 carbon, which isn't enough for a hexose sugar. Hexose means 6. So the Calvin cycle has to happen 6 times before a hexose sugar is made. And that could be glucose, it could be sucrose, and then the plant can use that glucose or sucrose and convert it into whatever organic compound is needed. So that could be another carbohydrate, so cellulose for the cell walls. It could be a store of glucose in the form of starch, or it could convert the hexose sugar into glycerol and then combine with fatty acids to make lipids, or it could combine it with nitrates to form the amino acids. Now you need to know a little bit about limiting factors as well, and the limiting factor is anything that reduces the rate of photosynthesis, and that could be light intensity, carbon dioxide concentration, or temperature. And the way you can look at a graph to tell what is currently limiting the rate of reaction is wherever you have a positive correlation on the graph, that tells you that whatever is on the x-axis is currently limiting the rate. So that means up until this point, or probably a bit further up to here, light intensity is limiting the rate of reaction. And we know that because as we increase the light intensity, the rate increased. But that then starts to plateau and level off. So even though you add more light, the rate doesn't increase. And what that means is there must be another factor that is now limiting the rate. And that must be either carbon dioxide concentration or temperature. So it's the same concept with carbon dioxide concentration, that as the rate is increasing and the carbon dioxide concentration is increasing, we know that carbon dioxide is a limiting factor until it plateaus. Temperature, this is linked to the idea of enzymes because it's an enzyme-controlled reaction. So at low temperatures, the rate of reaction is lower because we don't have enough successful collisions, there's not enough kinetic energy, we don't get enough enzyme-substrate complexes. But if it gets too hot, the enzyme denatures. Now the carbon dioxide, if you had to say why that limits, that is because it is a reactant in the Calvin cycle. Light intensity can limit the rate of reaction because light energy is needed in the light-dependent reactions for photolysis and photoionization. So for maximum photosynthesis, in agriculture, when they're growing crops, they need to consider how they can remove those limiting factors to maximise their profits. So sometimes you get application questions linked to this. So the idea of adding in artificial lighting, heat in a greenhouse, and that could be through burning fuels because that will also produce carbon dioxide. But all of those additions cost. So it has to be considered whether the cost of the additional light, heat or fuel is offset by the additional profit. So they have to make sure it's cost effective in order for it to be worthwhile to pay for those additional conditions. Next then we have a look at respiration, starting with aerobic respiration. And there are 4 key stages: glycolysis, the link reaction, Krebs cycle and oxidative phosphorylation. And for each stage, you need to know exactly where in the cell it occurs. And that's what we have in brackets here. So we'll start with glycolysis. This does produce some ATP, but not a lot. And it also happens in anaerobic respiration because oxygen is not required for glycolysis. It happens in the cytoplasm, not actually inside of the mitochondria. And there are 3 key stages that AQA say you need to know. First thing that happens is that glucose is phosphorylated to glucose phosphate, and that uses 2 molecules of ATP. That glucose phosphate very quickly is converted into triose phosphate, and then triose phosphate is oxidated to form pyruvate, and that produces 4 ATP and reduced NAD, which is the coenzyme used in respiration. So let's just have a look at what that would be on a diagram. Again, the yellow circles are representing carbon. So glucose is a 6-carbon compound. We said the first thing that happens is glucose is phosphorylated, and what that means is phosphate groups are added to it. Now we're going to add 2 phosphate groups, so we need 2 ATP molecules, and that creates glucose phosphate. That is an unstable compound, so it splits into 2 3-carbon compounds called triose phosphate. And then the triose phosphate, or TP, both of those molecules are oxidized. Both TP molecules will result in the gain of 2 ATP when pyruvate is made, and also as each is oxidized, there'll be a reduced coenzyme created. So NAD picks up a hydrogen to form NADH, or reduced NAD. So at the end we have 2 molecules of pyruvate, a net gain of 2 ATP, and what we mean by that is, although we've made 4 ATP molecules in this stage, we had to use 2 at the beginning. So overall, we've created 2. We've also made 2 reduced NAD. So that pyruvate that has been created and the reduced NAD, they are needed in the next stages, which is within the mitochondrial matrix. So they have to be actively transported into the mitochondrial matrix, ready for the link reaction. So the pyruvate is going to be oxidized further to make acetate. We're going to have an NAD picking up a hydrogen to create yet another reduced NAD molecule. So that's the first step we can see here. Pyruvate, our 3-carbon compound, it's going to be oxidized further, and we can tell it's been oxidized because it's lost the hydrogen, which has been picked up by the coenzyme NAD. So that has created acetate. Acetate is only a 2-carbon compound, and that is because carbon dioxide is produced at this stage of the link reaction. Now that's not the end, because the acetate then combines with another coenzyme called coenzyme A, and that creates acetyl-CoA. And acetyl-CoA is what can then enter the Krebs cycle. So overall, for every glucose molecule, there are 2 pyruvates created. So for every glucose molecule, the link reaction happens twice. So for one glucose molecule, we'd create 2 acetyl-CoA, 2 carbon dioxides, and 2 reduced NAD. So that acetyl-CoA can then enter the Krebs cycle, and it combines with a 4-carbon molecule. And that will then create a 6-carbon molecule. Now the coenzyme, its only job is to deliver the acetate to that 4-carbon compound. So once that happens, the coenzyme is released and it can be reused in the link reaction. We then have a series of redox reactions, and the Krebs cycle is going to be generating even more reduced coenzymes, a small amount of ATP, and some carbon dioxide. And that's what we can see in this stage here. The 6-carbon to the 4-carbon molecule, which is then regenerated. To go from 6 to 4, we must have lost 2 carbons, and that's in the form of 2 carbon dioxides. We only have 1 molecule of ATP being produced, but we have quite a few reduced coenzymes. We create 3 reduced NAD and 1 reduced FAD, which is our final coenzyme involved in respiration. So overall, the products per cycle are 3 reduced NAD, 1 reduced FAD, 1 ATP, and 2 carbon dioxides. But for every glucose, the cycle happens twice, because we'd have 2 acetyl-CoA produced, so 2 rounds of the cycle. So we actually double all of those values. So look carefully at an exam question, whether they're asking for the products per cycle or per glucose molecule. Oxidative phosphorylation is the final step, and this is very similar to what we looked at in chemiosmosis in the light-dependent reaction of photosynthesis. It's when most of the ATP is produced. So the first thing that happens is, in the mitochondrial matrix, all of those reduced coenzymes that were produced are going to release their hydrogen, and that splits into protons and electrons. Those electrons are going to be transported along the electron transfer chain. That releases energy, and that energy is used to transport the protons from the mitochondrial matrix into the intermembrane space. That creates this electrochemical gradient, and therefore the protons can move by facilitated diffusion down their concentration gradient through ATP synthase. And the ATP synthase will then phosphorylate ADP to create ATP. So that is how we create lots of ATP in this stage, and you actually create 34 ATP molecules. Now at the end of the electron transfer chain, those electrons are picked up by oxygen, and oxygen also picks up the protons once they've passed through, and that is how we create water. So we say that oxygen is the final electron acceptor, and it will form water when it also picks up those protons. Anaerobic respiration is without oxygen, and it only occurs in the cytoplasm. In plants and microbes, anaerobic respiration produces ethanol and carbon dioxide. In animals, it produces lactate or lactic acid. And this stage is all to do with reoxidizing NAD so that glycolysis can continue to occur. So what we have happening is glycolysis occurs as usual because no oxygen is needed for that stage. But instead of the pyruvate being transported into the mitochondrial matrix for the link reaction, it remains in the cytoplasm, and the pyruvate is reduced to form lactate. We know it's reduced because the reduced coenzymes that were just produced in glycolysis are immediately reoxidized to NAD, and that is because the pyruvate gained the hydrogen. Now that is important because that NAD can then be reused in glycolysis so that glycolysis can continue to occur. And that ensures that at least some ATP is continually made. So the downside is lactic acid is acidic, so it can denature enzymes and other proteins, but that is the price to pay so that at least some respiration can still occur. But you can't respire anaerobically for long because that buildup of lactic acid will eventually denature the enzymes involved in glycolysis. Same idea for microbes, except we have ethanol and carbon dioxide produced. Again, ethanol is going to be harmful, but it's to ensure that glycolysis can still continue, so at least some ATP is being produced for metabolic reactions to continue. The next bit in this topic is looking at how energy is transferred in an ecosystem. So it's looking at food webs. And all food webs begin with producers, which are green plants, because they can photosynthesize to use the light energy to make their own organic compounds. So they don't need to eat, they produce their own food. Between each trophic level, which is the term of what we call each stage in a food web, the majority of energy is lost, and that is due to respiration and excretion. The remaining energy is used to form the biomass. And the biomass is the mass of carbon within the organism, or you can describe that as the dry mass. Now that is a key term, so definitely learn that definition. And we give biomass per given area, so it can be a fair comparison between organisms. MPP and GPP are two key terms that you need to know. So how productive an ecosystem is depends on the abiotic and biotic factors that are within that ecosystem. If there's plenty of water, light, it's warm, there's lots of green plants, then you'll have a really high rate of photosynthesis, and that results in more carbohydrates being produced by the plant. And we can quantify this using GPP and MPP. So GPP is gross primary production, and it is the chemical energy stored in the plant biomass in a given area, or it could be volume, and it's the total energy resulting from photosynthesis. NPP is the net primary production, and that is the GPP minus the energy that will be lost by respiration. So basically, MPP is the energy you have left to go towards making the biomass. Another formula that you need to be aware of is this one here, and this is looking at the net production of consumers. So to work this out, which is represented by the letter N, for organisms such as animals, you'd use the formula below, where N is the net production of the consumers, I is the chemical energy stored in ingested food, F is the chemical energy lost to the environment in feces and urine, and R are the respiratory losses. So here's the formula, and it's come up a couple of times as just a multiple choice question, just ticking which is the correct formula. So that's a good one just to learn, maybe put on a flashcard. So here's just a visual representation. We're basically looking at the amount of energy going in minus the energy losses coming out, which is the energy lost in respiration and the energy lost in feces and urine. The rates of productivity are recorded using the units kilojoules per hectare per year. So there's 3 parts to those units, and it can be asked as a 2-mark question to give the units or to say why you have 3 parts to the units. So the kilojoules is the unit of energy, but these units also include per unit area and per year. So it's recorded per unit area to standardise the results to enable environments to be compared. So it's taken into account that different environments will vary in size. The units are also per year to take into account the impacts of the seasons. So when you have rain, when it's light or when it's less light, the temperature changes. That's going to affect the productivity because it affects photosynthesis. So we do it per year, so you have an annual average, so it's a fair comparison. The last thing within this topic is the nutrient cycles. And we're going to start with the nitrogen cycle. The nitrogen cycle is really important because nitrogen is within proteins, ATP and nucleic acids. So that's the DNA and RNA. So it's essential that animals, plants, microbes have nitrogen within them to create these molecules. And we need this cycle because you can't actually gain nitrogen gas through gas exchange, like you can with oxygen or carbon dioxide for plants for photosynthesis. And the reason for that is nitrogen gas has this triple bond, and we don't have the enzymes to be able to break those bonds and then use the nitrogen in compounds. Microorganisms do though, so they have the enzyme to be able to do that, or some of them do, and therefore we need this cycle involving microorganisms to make the nitrogen accessible for nitrogen containing substances, which plants and animals can then use. The nitrogen cycle involves these key processes: ammonification, nitrification, nitrogen fixation and denitrification. But you also need to be aware that saprobiotic nutrition and microbes are essential for all of those stages highlighted in blue. So saprobiotic nutrition is referring to when microbes are feeding on dead plant matter or waste, and it's the microbes that are going to be doing that. So if we have a look at the cycle, we have nitrogen gas in the atmosphere. There'll be nitrogen fixing bacteria that can convert that nitrogen into ammonia. And that will either be done directly by nitrogen fixing bacteria in the root nodules of leguminous plants, which are types of plants like clover and beans, which have these lumps or nodules on their roots that contain bacteria. So for them, the bacteria will directly convert the nitrogen in the atmosphere into ammonium into their roots. Plants that don't have those root nodules, there is free living nitrogen fixing bacteria within the soil, which can also fix nitrogen into ammonium. Then we have nitrification occurring, which is when nitrifying bacteria can oxidize ammonium into nitrites and then oxidize it further into nitrates. And that can then be absorbed by active transport into the plant roots. And then animals can absorb that through digestion and absorption. Now, the decomposers will be breaking down the nitrogen containing compounds in the dead plant and animal material and in the waste produced by animals. But for A-level, it's not enough to just say the dead material or waste. You have to name the nitrogen containing compounds. So the waste, you could name urea. And then in the dead plant and animal material, it would be proteins. It could be DNA as well. So you have to name the compound that the decomposers would be breaking down. And this would be our saprobiotic nutrition. So the decomposers will break down the proteins to release the nitrogen, and it converts it back into ammonium, and then the cycle can go round again. If, though, there is a lack of oxygen within the soil, we then have anaerobic denitrifying bacteria taking over, and they can convert the nitrates back into nitrogen gas. So that's how we get the nitrogen going back into the atmosphere. So other than the denitrifying bacteria stage, all of the other stages are oxidation, and they would require oxygen to be present. The other cycle you need to know is the phosphorus cycle. And phosphorus is really important because it's used in the phosphate groups of DNA, RNA, ATP, and in the phospholipids within the phospholipid bilayer. So it is an essential element for life for all organisms. The phosphorus cycle is different to the carbon and nitrogen cycle. Not that you actually learn the carbon cycle at A-level. And that is because you do not find phosphorus as a gas in the atmosphere. So instead, the main store or source is as phosphate ions in the mineral form in sedimentary rock. Now you need to know about the mycorrhizae, which are fungal associations which occur between the plant roots and beneficial fungi. Now, although we're talking about this here in the phosphorus cycle, it would also be occurring in the nitrogen cycle as well. The mycorrhizae are going to provide benefits for plant growth. And that's because the mycorrhizae fungal associations, we can see here you get these really long networks of fungi, and they can spread out in the soil to create a larger surface area for the absorption of water and mineral ions going into the roots eventually. The plants provide an advantage to the fungus because the plants through photosynthesis will create carbohydrates, and it can share those carbohydrates with the fungus. So this is a mutualistic relationship because they both provide a mutual benefit to each other for survival. So the phosphorus cycle, if we start in the middle looking at the phosphate ions in the oceans, or it could be within the soil, plants can absorb those phosphate ions through their root hair cells by active transport. Animals will then be eating the plants so they can consume and absorb the material to get the phosphate ions. When animals excrete, that will release phosphorus back into the oceans or the soil in the form of phosphate ions. As well as returning it to the soil, we also have decomposition. And phosphate ions from waste and remains are going to be broken down. So animals which have a shell, but also bones, contain phosphate ions. And those can be eroded to release the phosphate ions back into the ocean or the soil. The other thing you can have, which we can see in this image, is guano. And guano is from bird feces, and it's this white layer you can see on the rocks. So it's a type of excrement which is really rich in phosphate ions. Deposition is when we get phosphate ions building up to create the rocks, and that also links to sedimentation over here. So over time, from high pressure as well, the phosphate ions within the oceans, within the soil, will compact through sedimentation to create the sedimentary rocks. And then the last link back is the fact that those sedimentary rocks can be eroded to release the phosphate ions back into the soil or oceans. Now, the extra one added here is the idea of fertilizers. Fertilizers added to soils can also run off from the soil or leach into water sources, so it would return it back into oceans, or it could just be within the soil itself, but that will be adding phosphorus into the cycle. If we just look at fertilizers a little bit further, we said that they can be added to the soil. The reason for that is when crops are harvested, you're removing the plants, and that is therefore removing one of the stores of nitrate or phosphate ions. So it's going to be taken out of the nutrient cycle. And if you continue to do that, eventually you'll have plants and soil which are mineral deficient. So you have to add either natural or artificial fertilizers to replace the minerals and the nutrients that are being removed when you harvest crops. So natural fertilizer would be animal manure. Artificial is soluble chemicals which contain exactly the proportion and quantities of the mineral ions required. Now, the natural fertilizers are cheaper because if you have your own animals on a farm already, it's completely free. It is just the manure from the animals. However, it doesn't contain necessarily the exact proportions of the minerals you need, and you have no control over that. Artificial fertilizers, because they are synthetically created, they are created to have the exact proportion of minerals that plants would require. However, the inorganic substances are very, very soluble, which has the downside of that they can dissolve in the rainwater and leach, which means they're washed away. So those soluble chemicals such as nitrate, phosphorus, dissolve in the water and then they leach, which means they run off from the rainwater into nearby water sources, such as rivers or ponds. If that nitrogen fertilizer leaches into waterways, it causes eutrophication. And eutrophication is when nitrates are going to stimulate the growth of algae in a pond or a river. It's more likely to happen in a pond though, because in a river, the movement of the water can prevent the algae from growing or staying in one place. So the excessive growth of algae creates this blanket on the top of the water. That blocks the light. As a result, plants below that blanket aren't going to be receiving light for photosynthesis and they end up dying. Bacteria within the water will then feed and respire on the dead plant matter, and in doing that, they're going to be using up the oxygen within the water. And you get more and more bacteria growing and thriving because of all that dead plant material to feed on, so even more oxygen is used up. And eventually there won't be enough dissolved oxygen within the water for animals, and animals such as fish and other aquatic organisms will therefore die because there's no oxygen for respiration. We begin with one of the key definitions, and that is of the term stimulus. So a stimulus is a detectable change in the environment, and the cells that can detect these changes are called receptors. So organisms increase their chance of being able to survive if they can respond to these changes in the environment. And we look at in this topic the different mechanisms for how organisms do this. So we begin with the responses in flowering plants. And tropism is the term given to when a plant responds to its surroundings via growth. Now, tropisms can be described as either positive or negative. Positive meaning the plant grows towards the stimulus, negative meaning it grows away. And the two stimuli that you need to know are light and gravity. Now, tropisms are actually controlled by specific growth factors, and one key example is IAA. Now, IAA is a type of auxin and it can control cell elongation, meaning making the cells get longer. And this is in shoots, it elongates, but having IAA in the roots actually inhibits the growth of cells. IAA is made in the tip of both the roots and the shoots, but it can move through by diffusion to other cells. Phototropism is the term given to the tropisms where the plant is responding to light. And for shoots, the reason that it improves their survival chance if they respond to light is because light is needed for the light-dependent reactions in photosynthesis. So plants' shoots will grow and bend towards the light source, and this is positive phototropism. Now the shoot tip cells, as we already said, those will be producing IAA, and that causes cell elongation, causing the shoots to grow upwards towards the light source. Now the IAA diffuses to the shady side. So in the first images we can see that the IAA is diffusing downwards away from the light source. However, in the other images we can see we've got a unilateral light source, meaning it's coming from one side. And picture 3 shows you the light to the right hand side. And the IAA will still diffuse to the shady side, that just happens to be on the left. And that will cause the cells on that side to elongate, and therefore the shaded side cells will be stretching and that side will be longer, and that causes the plant to end up bending towards the light source. In the roots we have the opposite happening. The IAA will still diffuse away from a light source, but this time we said that in the roots IAA inhibits the growth. So as a result, the top layer of cells will be growing or elongating more, and that is why the root ends up bending downwards away from the light. And this helps survival because that will help anchor the plant further into the ground and potentially reach more water sources. So that's an example of negative phototropisms. Gravitropism is the term given for the responses to gravity. And in the shoots, the IAA will diffuse from the upper side to the lower side of a shoot. So it's diffusing down towards gravity. And if a plant shoot was vertical, then that would mean the IAA would be diffusing down, causing the lower cells to elongate, and the plant would be growing upwards. Now the image we can see down here is if a shoot was on its side, and that will cause the shoot to bend upwards, and that's because the IAA diffuses down, the cells on the lower side will elongate, and that is why it's bending up. And this is against gravity, so that would be an example of a negative gravitropism. Now in the roots, IAA moves to the lower side of the roots also, and that would cause the upper side to elongate and the root will bend down towards gravity. And that would be a positive gravitropism. And that increases survival because again it helps to anchor in the plant. Now a reflex is an example of a rapid automatic response to protect animals from danger. A reflex arc is made up of 3 neurons, and that would be the sensory neuron. We'd then have the relay neuron and the motor neuron. So it's only 3 neurons. That means you're only going to have 2 synapses, and that is one of the reasons why it's such a rapid response. Now, some more simple responses are taxis and kinesis, and these keep organisms within their favourable conditions within their environment. In particular, we're thinking about light, moisture, and potentially chemicals as well. Taxis is one of these types of responses, and this is when an organism will move its entire body towards a favourable stimulus or away from an unfavourable stimulus. So the example we have here is an earthworm, and earthworms don't want to be in the light. That would dry them out. So if it is light, they would move their entire body away towards a darker light source. So when an organism moves towards a stimulus, this is known as positive taxis. When an organism moves away, it's described as negative taxis. So the earthworm moving away from the light would be negative phototaxis. Kinesis is slightly different. This is when an organism changes the speed of movement and the rate that it changes direction. So if an organism moves from an area where there are beneficial stimuli in the area within the harmful stimuli, its kinesis response will be to increase the rate it changes direction, and that's to increase the likelihood that it will return itself to the favourable conditions. If an organism, though, is surrounded by the negative stimuli, the rate of turning decreases to keep it moving in a relatively straight line, and this will actually increase the chances of it moving to find a new location with the favourable conditions. Receptors we talked about at the start. So these are cells which can detect a change in the environment or stimuli. And each receptor responds only to one specific stimuli. And this stimulus of a receptor can lead to an action potential or establishing a generator potential. And that is what results in an organism being able to respond. Now, there are 3 receptors that you need to know: the Pacinian corpuscle, rods and cones, which are both light receptors or photoreceptors found in the eye. So if we start with the Pacinian corpuscles, these detect changes in pressure, so they're pressure receptors, and you mainly find them deep in the skin of your fingers and feet. And this is what we're seeing here. This is a cross section through your skin, and the Pacinian corpuscles are about the same level, slightly lower than your hair follicles. Now, the Pacinian corpuscle is actually a sensory neuron wrapped in lots and lots of layers of plasma membrane. And that has special channel proteins embedded within those plasma membranes. So that's what we're seeing here. We've got our sensory neuron with a nerve ending. We've got lots of connective tissue with gel in between each of these layers. Now, the membranes of the Pacinian corpuscle have stretch-mediated sodium channels. So where we said it's special channel proteins, that's what we're referring to. And what this means is when these proteins have pressure applied to them, they are stretched and deformed, and it opens the channel. And in this case, it opens these sodium ion channels. So if pressure is applied to the finger, for example, the Pacinian corpuscles deep in the skin of your finger will be stretched and deformed. That will widen the sodium ion channels, and therefore sodium ions will be diffusing in, which leads to the establishment of this generator potential. So it's diffusing into the axon within the sensory neuron, and if enough sodium ions diffuse in to exceed that threshold, then you have an action potential and a response will occur. The next 2 receptors are both found in the human retina, and these were the photoreceptors, rods and cones. Now, rod cells are named because of their shape. They are more rod-like in shape, and they can only process images in black and white. Now, to create the generator potential, the pigment in the rod cells, which is rhodopsin, must be broken down by light energy. Now, they can detect light at very, very low intensities. And the reason for this is many rod cells connect to one sensory neuron, which we call retinal convergence. Now, although this gives the benefit that you're able to see in black and white, even if it's very dark, because all of those connecting rod cells contribute towards reaching that threshold to generate an action potential, the downside is it provides low visual acuity. And what that means is it's difficult for the brain to distinguish between the separate light sources. So you don't have very good clarity in your vision. So that's why at night time, if you do happen to wake up, you'll be able to see, but it'll only be black and white, and you won't be able to see particularly clearly, which is why you might end up bumping into things if you try and walk. Now, the other type of cell that is a photoreceptor is the cone cells. And there are 3 types of cone cells, and each one contains a different type of iodopsin pigment. There's one that is for red, green, and the last one blue. And they all absorb slightly different wavelengths of light. And we don't just see red, green, and blue in our vision, but depending on the proportion of red cone cells or green or blue cone cells that are detecting a stimulus, that will give the different colours, not just red, green, and blue. Now, iodopsin is only broken down if there is a high light intensity. So you can only see in colour if there is enough light present. And the reason for that is we only have one cone cell connecting to one sensory cell. And therefore, there has to be a high enough light intensity to break down enough of the iodopsin pigment to be able to trigger an action potential. Now, that gives the property of high visual acuity. So because only one cone cell is connected to one sensory neuron, that means the brain can distinguish where the separate source of light was detected, and therefore you have a lot more clarity and precision in your colour vision. Now, the distribution of the rods and cone cells is not equal across the retina. You actually have most of the cone cells located in this part here called the fovea. And that is where you have the highest light intensity reaching the retina, because that is where the light is focused by the lens. Now, that is an advantage because the cone cells will only be able to detect the colour lights in high light intensities. And that's why you mainly find them at the fovea. The rod cells can still detect light even at low light intensities, and that is why they are found further away, where there'll be slightly less light being able to reach that part of the retina. Now, the next thing you need to know about is how your heartbeat is controlled. Cardiac muscle is myogenic, and what that means is it will contract and relax on its own accord without any nervous system input. However, the speed at which it contracts is controlled by the nervous system. And that is what you need to know about in this section of the A-level. Now, the parts that are involved in controlling the heart rate are, first of all, the sinoatrial node, or the SAN. And this is located in the right atrium. This group of cells or tissue is known as a pacemaker, and that's because they set the pace at which your heart will beat. You then have the AVN, or the atrioventricular node, which is located between the right atrium and the left ventricle. The bundle of His is what we call the tissues running down the septum. And then you have the Purkinje fibres, which branch through the ventricle walls. Now you can see it on the outside here, but it'll be branching all the way around the ventricle walls. Now, how those all work together to control the heart rate is linked to what you would have learnt in the topic 3 when you did the cardiac cycle. So when you learnt about atrial systole and atrial dystole, or diastole and systole, people say it differently. So the SAN starts off this process by releasing this wave of depolarization or electricity, and that will spread across the two atria. And when the wave of depolarization hits the muscle, it causes it to contract. So that is why the atria contract first. When that wave of depolarization reaches the AVN, that causes the AVN to release another wave of depolarization. Now, there's actually a non-conductive layer of tissue that separates the atria and ventricles, and that prevents the wave of depolarization travelling straight down to the ventricles. So instead, what happens is that wave of depolarization that was released by the AVN travels down the bundle of His and then up through the Purkinje fibres. Now, because of that, it causes a very slight delay in the time it takes for the apex of the heart, so the base of the ventricles and the ventricle walls to contract. And that is actually an advantage because that means it gives the atria enough time to contract and force all the blood into the ventricles before the ventricles will then contract. And the ventricles would contract from the apex first and then move upwards. And that's an advantage because it's a bit like squeezing toothpaste out of your toothpaste tube. If you squeeze from the bottom upwards, you're going to get all the toothpaste out. And it's the same here. Squeezing and contracting that cardiac muscle from the bottom, then upwards, forces all of the blood out of the heart. Finally, all of the cells repolarize, and that causes the cardiac muscle to relax, and then the cycle happens again when the SAN releases another wave of depolarization. Now, the involvement of the nervous system is what triggers the rate at which the SAN will release those waves of depolarization. And the parts involved are the medulla oblongata in the brain. That is what controls the heart rate, but this is through the autonomic nervous system, meaning it's automatic and there's no conscious control. So you can't think and control your heart rate. There are two key parts, and that is the sympathetic nervous system and the parasympathetic nervous system. And that's what we're being shown here, these two key routes that the impulses can take connecting the medulla oblongata to the SAN in the heart. Now, the sympathetic nervous system, if impulses travel via the sympathetic nervous system, that will cause the SAN to release the waves of depolarization more frequently and therefore increase the heart rate. If more impulses are sent down the parasympathetic nervous system to the SAN, that will cause the SAN to release these waves of depolarization less frequently, and therefore it decreases the heart rate. One of the stimuli that will change the heart rate is pH, and the other one is blood pressure. There are different receptors that detect these different stimuli. The changes in pH are detected by the chemoreceptors, and the blood pressure changes are detected by pressure receptors. But they are both found in the same location. You find them in the walls of the aorta and also the carotid artery. And this is the next blood vessel that attaches onto the aorta. Now, your blood pH does change, and during times of high respiratory rate, say for example maybe if you're doing lots of exercise, the blood pH will decrease. And that is because of the excess carbon dioxide from aerobic respiration or lactic acid from anaerobic respiration. And these acidic compounds have to be removed because if you have excess acid in your blood, it can denature enzymes. So this is achieved by increasing your heart rate so that the dissolved carbon dioxide in your blood can reach your lungs faster and therefore diffuse out into the alveoli and be exhaled. So that would mean you need more impulses going down the sympathetic nervous system to the SAN to increase the heart rate. Now, for blood pressure, if it's too high, it can cause damage to the walls of the artery. So it's very important to put mechanisms in place to bring down your blood pressure. And to do that, you would have more impulses sent via the parasympathetic nervous system to cause the SAN to fire less frequently and therefore decrease the heart rate. If your blood pressure is too low, though, that might result in insufficient oxygenated blood being supplied to respiring cells. So that would mean that your heart rate needs to be increased to try and counteract this. So therefore, the pressure receptors would detect this and result in more impulses being sent via the sympathetic nervous system to the SAN to cause the SAN to fire more frequently and therefore increase the heart rate. Now, if we have a look at a myelinated motor neuron structure, the key parts are, first of all, the cell body. And this cell body contains all the organelles you'd have in a typical plant cell. So, for example, we can see the nucleus. It also has proteins and neurotransmitter chemicals which are made in that part of the cell. The dendrites we can see here branching carry action potentials to surrounding cells. The axon is the conductive long fibre that is making up this whole part of the cell, and that carries nerve impulses all the way along this particular motor neuron. Schwann cells are what we call these layers wrapping around the axon, and they form the myelin sheath, which is a lipid, and therefore it insulates. It does not allow the charged ions to pass through to the axon. There are gaps though, and these gaps are called the nodes of Ranvier, and that is where they are not insulated. A resting potential is what we would have happening inside of the axon when there is no stimulus. So when a neuron is not conducting an impulse because there's no stimulus, there's no change in the environment, there is still a difference between the electrical charge inside the neuron, so inside the axon, and outside. And we call that difference the resting potential. There are more positive ions, the sodium ions and potassium ions, outside compared to inside of the axon. And therefore, the inside of the neuron is comparatively more negative. And that is why the resting potential is minus 70 millivolts. Now, you need to know how that resting potential is established in the first place after a response to a stimulus, and how it's also maintained if there is no stimulus. Now, the key way it's maintained is by a sodium-potassium active transport pump. So this is an example of co-transport, but it is also active transport because ATP is involved. And at this pump, you have 2 potassium ions being pumped into the axon, and 3 sodium ions are being actively transported out of the axon. This creates an electrochemical gradient, and therefore the potassium ions, because there's a higher concentration of them inside the cell, they will start to diffuse out of the cell. The sodium ions, there's a higher concentration outside, so they will start to diffuse into the cell. However, the membrane is more permeable to potassium ions because there are more potassium ion channels and because some of those ion channels remain permanently open. So that means more potassium ions can diffuse out than sodium ions diffusing in. And as a result, there are more positive ions outside of the cell compared to inside of the cell, and that's how we get the minus 70 millivolts. Now, if there is a stimulus, and if it's a large enough stimulus, it can generate an action potential. An action potential is when the neuron's voltage increases beyond a set point from the resting potential. And this is what generates the nerve's impulse to then travel along the neuron. So an increase in voltage, also known as depolarization, is due to the neuron potential becoming more permeable to sodium ions and therefore becoming more positive. Once an action potential is generated, it moves along the axon like a Mexican wave. So let's have a look at then how this action potential is generated. A stimulus provides the energy that can cause the sodium ion voltage-gated channels in the axon membrane to open, and this will cause the sodium ions to start diffusing in. And that's what we can see here. One of those channels has now opened, and therefore sodium ions will start to diffuse into the axon. The potassium ion channel here is permanently open, but because we now have some sodium ions moving in, we start to see this increase in the voltage. Now, if the stimulus is large enough to cause enough of these sodium ion channels to open, then you'll have enough sodium ions diffusing in to cause an increase beyond the threshold potential. And if you do have an increase beyond that minus 55 millivolts, then you will always have the rest of this curve happening. You'll always have an action potential generated. Now, there are even more voltage-gated sodium ion channels. So as that voltage will start to increase, that will cause more sodium ion channels to open. Then we have this positive 35 to 40 millivolts. What then happens though is we see this decrease, and that's because when it does reach this particular voltage, it then causes the voltage-gated sodium ion channels to close. So no more positive ions are entering, but we still have those potassium ions moving out. So we start to get this decrease, which we call the repolarization. That will then cause more potassium ion channels to open, so even more potassium is diffusing out. And eventually, so many positive ions have diffused out, we get to that resting state, but actually it overshoots and goes to minus 80 millivolts, which we can see here. We're now back to that original state of potassium ions would be able to move out, but all of the other gates are closed. So that is the generation of an action potential. And this happens along the axon multiple times. This graph is not a change over distance. So this isn't happening just once along the axon. It happens right at the start of the axon, and then at every node of Ramvir, if it is a myelinated neuron, you'll then have another action potential being generated until you get to the end of that neuron and then a synapse. So it's a bit like the idea of a Mexican wave moving along. If it was unmyelinated, then same idea, but it has to happen at every single position on the axon because you wouldn't have the myelin sheaths, so that would mean it would take longer for all of those action potentials to finally reach the end. Now you need to know about this all or nothing principle, and it links to the threshold. So if the axon isn't depolarized to at least minus 55 millivolts, which is the threshold to cause the rest of the action potential to be generated, then the impulse will not be produced. So what we mean by the nothing part of the principle is, if the stimulus isn't large enough to cause a voltage increase to at least minus 55 millivolts, then nothing more will happen. And that's what this bit is showing you here, some failed initiations of an action potential. The all part of the principle is the fact that if you do exceed that threshold, then you will always have an action potential, and it will always peak at exactly the same point. So even if the stimulus is bigger, so brighter light compared to dimmer lights, you will still have the same peak. But what changes is the frequency of those action potentials being generated. So that is the all or nothing principle, and the reason it's important is to make sure that animals are only responding to stimuli which are large enough rather than responding to every single slight change in the environment, because if we did that, it would actually overwhelm your senses and it wouldn't necessarily be protecting you from stimuli which might be causing you danger. Now, the refractory period we mentioned is when it goes into this minus 80 millivolts. But the importance of this is, once we have this stage, we have a period when another action potential can't be generated straight away. And that's because these sodium ion channels, we describe as they're recovering. But essentially what we mean is, because we've now gone even more negative, you would have to have even more sodium ions to be able to reach that threshold. The importance of this is, first of all, it ensures that discrete impulses are produced. So an action potential cannot be generated immediately after another, and that makes sure each one is separate. So that helps with you being able to identify the source of the particular stimulus. It also makes sure that the action potentials will only be travelling in one direction along the axon. So it stops the action potential from spreading out in both directions, which would actually result in the impulse not travelling along. It limits the number of impulses as well, and this is important to prevent you overreacting to a particular stimulus or over-responding. Now, the factors that will affect the speed of conduction are myelination and saltatory conduction, the axon diameter, and temperature. So the first one was myelination, and that results in saltatory conduction. And what that term means is the action potentials will jump from node to node, meaning the node of Ranvier. And that is because this myelin sheath created by the Schwann cells is insulating, so the ions cannot diffuse in and out during these sections, so it jumps to the parts where there is no insulation. Now, as a result, that means that the impulse will travel much faster along the axon because you don't have to generate as many action potentials. You're only generating them at those nodes. Next then, the axon diameter. The wider the diameter, that is going to increase the speed. And the reason for that is basically like the idea of physics and wires and electricity. So the wider the diameter, that means we're going to have less leakage of the ions, and therefore the action potential can move faster. Temperature, there's two reasons why this has an effect. Number one, because if you have a higher temperature, the ions will have more kinetic energy. That means they'll be able to diffuse faster. The second reason is the enzymes involved in respiration to provide ATP for the sodium-potassium pump will be able to work faster as well if there is a higher temperature. Now, if it does exceed the optimum of around 37, then that could actually decrease the speed. So synapses are the gaps that we have between the neurons. So once you have your action potential reaching the end of a neuron, it has to get to the next one. And in order to do that, we have to have neurotransmitters, which are chemicals, diffusing across the gap, which will then generate the action potential in the next neuron. Now, the way that this happens is, first of all, your action potential, as I said, it will arrive at the end of the neuron, and we describe that as the synaptic knob. Depolarization of the synaptic knob leads to calcium ion channels opening, and therefore calcium ions will diffuse into the synaptic knob. Those calcium ions will cause the vesicles that contain the neurotransmitter to move towards the membrane, fuse, and release the neurotransmitter into the synaptic cleft. Because we have a high concentration of the neurotransmitter where they've just been released, they'll then diffuse across to the postsynaptic membrane. And on the postsynaptic membrane, there are receptors, complementary in shape, for those neurotransmitters to bind to. When those neurotransmitters bind to the receptors, it causes the sodium ion channels on the postsynaptic membrane to open. Therefore, sodium ions will diffuse in, and if enough sodium ions diffuse in to exceed that minus 55 millivolt threshold, then the action potential is generated. So that will then cause that action potential to be generated in the postsynaptic membrane, and then it can continue along the axon of the next neuron. Now, this is unidirectional, and you could be asked, how is it ensured that it's unidirectional? And there's a couple of ideas. Number one, the vesicles containing those neurotransmitters are only in the presynaptic neuron, so they can only be released from this side. Because of that concentration gradient, diffusion happens from the pre to the postsynaptic neuron, and you only find the receptors complementary in shape to those neurotransmitters on the membrane of the postsynaptic neuron. Now, one example of a synapse that you need to know is the cholinergic synapse. So it's exactly the process that we just went through, but you need to know the name of the neurotransmitter. So the neurotransmitter is acetylcholine. It will bind to its receptor, and then it will go through the whole stages that we said. But those neurotransmitters don't remain permanently bound. If they did, that would mean the sodium ion channels would be continually open, and you'd continually be triggering this action potential and a response, even though the stimulus wasn't present anymore. So to prevent that happening, there is an enzyme that will break down the acetylcholine into choline and acetate, which can then be reabsorbed into the presynaptic neuron and be reused. And that neuron is acetylcholine esterase. Now, I said that in order for the action potential to be generated in the postsynaptic neuron, you needed to have enough sodium ion channels opening, and that is caused by the neurotransmitters binding to the receptor. And there's two different mechanisms of summation to ensure that happens. Summation is referring to the fact that you have this rapid buildup of neurotransmitter to generate the action potential, and you can either have spatial or temporal. Spatial is when you have multiple neurons connecting to the same postsynaptic neuron. And collectively, these three neurons in this example should release enough neurotransmitter to cause enough of those sodium ion channels to open on the postsynaptic neuron. The alternative is temporal, and this is where you only have one presynaptic neuron connected to one postsynaptic neuron, but instead the way that this will cause enough neurotransmitter to be released is you have repeated releasing of that neurotransmitter over a short period of time, until eventually you have enough neurotransmitter bound to enough of the receptors on the postsynaptic neuron's membrane to open enough of those sodium ion channels. You also have inhibitory synapses, and in this case you would have chloride ions would be moving into the postsynaptic membrane. So you'd have different neurotransmitters binding to their complementary receptor, but this time it would cause chloride ion channels to open. And because you'd then have negative ions moving in, this would cause the membrane potential to decrease to at least minus 80 millivolts, and therefore the postsynaptic membrane would go into this hyperpolarization, making it very unlikely that an action potential could be generated. Neuromuscular junctions are what you have right at the end of your response arc. So you have your motor neuron, you then have a gap, and then you have your muscle. And your muscle is one of the effectors that would generate the response. And what you need to know is similarities and differences between the synapse that you find here at the neuromuscular junction compared to your typical synapse such as the cholinergic. So what they have in common is both of them will work in one direction only, so unidirectional. And that is because the receptors for the neurotransmitters are only found on the postsynaptic membrane, or in this case, the muscular membrane. A neuromuscular junction will only be excitatory, meaning it will only generate that response. Whereas a cholinergic, we looked at, could be excitatory, generating an action potential, or it could be inhibitory, which prevents the action potential from being generated. The neuromuscular junction will have a neuron connecting to a muscle, and it's always the motor neuron. The cholinergic is just a synapse between any two neurons. The neuromuscular junction is always at the end point for the responses, and the cholinergic synapse is always going to be generating a new action potential. For the neuromuscular junction, the acetylcholine will bind to receptors on muscle fibre, whereas the acetylcholine binds to receptors on the postsynaptic membrane in the cholinergic synapse. So that could be a long answer question, it could be a comparison table, so definitely learn those comparisons for those two. So that leads us on to muscles, which is an example of an effector, meaning it carries out the response. And muscles always occur in antagonistic pairs against the skeleton. And what that means is you have two different muscles that will be doing the opposite thing at the same time. So as one of them contracts, the other relaxes. Now this could be automatic or it could be conscious control for this movement. Now you need to know the structure of these muscles. So that's what we're looking at here. And we have the myofibrils labelled, and a myofibril is made up of lots of fused cells, and because they're fused together they will share organelles. So they share the nucleus, they'll be sharing the cytoplasm, and this cytoplasm, because it's all shared, is known as sarcoplasm. So there'll be lots of nuclei within one of these myofibrils, but there's also a high number of mitochondria, and that is because we'll be needing ATP from respiration for the muscle contraction. Now these myofibrils are all bound together to make up this muscle fibre. But what we're actually going to focus on is the myofibril in more detail, having a look at the sarcomere. Now the muscle fibres are made up of millions of myofibrils, which collectively bring about that force to cause the movement of the skeleton. And the myofibrils are made up of two key proteins, myosin and actin, and that is what forms the sarcomere. The sarcomere you can actually look at in terms of different sections or bands, depending on whether you're looking at a part which is just made up of the actin protein, the myosin protein, or where the two proteins overlap. And that's what this diagram is representing. The actin is shown in red, the myosin is shown in blue, and what you can actually see from the myosin is the position. You have these chains of myosin with heads sticking off, that's what these lines at right angles are showing, but also the myosin molecules are all bound together, and that keeps them in a fixed position. The actin you just have as a thinner filament, and they can slide closer together towards this M line. Now you could be asked to describe what happens to the length of the different bands when a muscle contracts. So the A band is the length of the myosin, so that should never change. The A band is constant because the myosin itself, apart from the heads, the actual length of the myosin is never going to move. The H zone is where you just have myosin with no actin overlapping. Now that will change when a muscle contracts, because when a muscle contracts, the actin is sliding in, so therefore the H band or H zone is going to decrease because that is where you have myosin by itself. The I band is also going to change in size because the I band is where you have just actin by itself. So when the muscle is relaxed, you have a bigger I band. When it contracts, the I band decreases because the actin has slid closer together, so you have more of an overlap with the myosin. You could be asked about what happens to the Z lines, and the Z lines are indicating the start and the end of one sarcomere. And because the actin slides in, the Z lines become closer together as well. Now, my tip for learning this is, it isn't really intuitive, the letters representing these bands, so I'd say put them on a flashcard. So A band on one side, on the other side just say it's the length of the myosin and it remains constant. The H zone is where you just have myosin by itself, decreases when muscles contract. So create a set of flashcards for this and just learn that content. This is just showing you the image as well of this happening under the microscope compared to the diagram. Now, the sliding filament theory is how the muscles actually contract. So when an action potential reaches the muscle at that neuromuscular junction, it's going to stimulate the muscles to contract. It does this by causing calcium ions to release from the sarcoplasm, and what this does is it causes the tropomyosin, which is the protein that covers the actin binding sites. It causes it to move out of the way, and therefore these binding sites are now exposed. Whilst ADP is attached to the myosin heads, which we can see here, the myosin head is able to bind to these exposed actin binding sites, and it forms what we call a cross bridge. The angle that this creates with the myosin head binding is going to result in some tension, and that causes this pulling or sliding action of the actin along the myosin. And we call that the power stroke, which we can see just here, when that head causes the actin to slide along. Now, in that power stroke action, the ADP and PI are released. ATP is the molecule that then binds to the myosin head and causes it to change shape slightly, and as a result, it detaches from the actin molecule. Within the sarcoplasm, there's an enzyme, ATPase, and that is activated by the calcium ions that were released in the sarcoplasm. So as long as you still have the stimulus causing these calcium ions to be released, then the next step will happen to trigger even more contraction. So that enzyme ATPase is going to hydrolyze the ATP that was attached to the head into ADP and PI. And that now releases energy for the myosin head to return back to its original position. And then we're back to the start again. That process repeats as long as the calcium ions remain high, the tropomyosin is moved out the way, and the myosin head combined. So this will repeatedly happen to slide the actin as close together so you have this full muscle contraction. So that continues to happen while the stimulus is causing that release of calcium ions. So you need to have ATP for this process to happen, but also a molecule called phosphocreatine. Now, if the muscles have been very active, we just saw there, it requires ATP. So the muscles require a high concentration of ATP to be able to contract. And the chemical phosphocreatine, which is stored in muscles, assists this process because it provides the phosphate. So in order to regenerate ATP, you need that inorganic phosphate to bind with ADP, and that's provided by the phosphocreatine. The last thing you need to know about muscles is two different types of muscles, slow twitch and fast twitch fibres, and you need to be able to compare their structure, location and general properties. So the slow twitch contain a large store of myoglobin, which is a type of hemoglobin which has a very high affinity for oxygen, and therefore it can hold on and store oxygen until the partial pressure is really low, and then it unloads. It also has a rich supply of blood, and that is so it can constantly provide oxygen, and many mitochondria, so you can have lots of aerobic respiration. So all of those structures link to the fact that these slow twitch fibres do contract slower, but they respire aerobically and for longer periods of time because of those structural adaptations. And that is for exercises which are going to be endurance, or it could be just regular exercises as well, just regular movements, nothing intensive. So for example, calf muscles when you're walking. Fast twitch fibre muscles are much thicker and there's more myosin filaments. They have a large glycogen store and a phosphocreatine store to help make the ATP. They also have a high concentration of enzymes needed for anaerobic respiration. Now, all of those structural features link to the properties of it contracts faster, short bursts of powerful contractions as well. So this is for intense exercise, and as we said, it'd be anaerobic respiration. And an example would be your biceps when you are contracting your biceps. So the last bit in this topic is homeostasis. And this involves physiological control systems to maintain your internal environment within set restricted limits. And the examples that you need to know are maintaining the core body temperature and maintaining the blood pH as well in relation to enzyme activity. Now, for those two, you don't really need to know much of the details. It's just knowing that those two conditions are controlled, and you'd need to know why in terms of the effect it has on enzyme activity. Homeostasis involves negative feedback, and this is when any deviation from the normal values, so if it goes too high or too low, there are mechanisms put in place to bring the conditions back to their original level. And it involves the nervous system and often hormones as well. Two key examples you need to know are blood glucose concentration and water potential. So we'll start with the blood glucose. Now, your blood glucose concentrations will increase after you have eaten food containing carbohydrates or drinks containing carbohydrates, and they'll decrease if you haven't eaten food containing carbohydrate for a while or if you have been exercising. The pancreas is the organ that detects these changes in blood glucose levels, and within the pancreas there are cells called the islets of Langerhans, which are responsible for releasing the insulin and the glucagon, which are the two hormones that bring blood glucose levels back to normal. Insulin is going to be released when blood glucose levels are too high, and that will bring blood glucose levels down to normal. Glucagon is released when the blood glucose levels are too low. So think of glucose gone, glucagon. And this will cause an increase in the blood glucose levels. Adrenaline, which is released by the adrenal glands, which are found just above the kidneys, this hormone is released when your body anticipates danger, and this results in more glucose being released from the hydrolysis of glycogen in the liver. Now, just to look at this as an overview, so your normal blood glucose levels, after you've eaten, they will increase. And this will be detected by the beta cells in the islets of Langerhans in the pancreas. The beta cells will release insulin. That will cause the liver cells to become more permeable to glucose, and there'll be enzymes activated to convert the glucose that's been absorbed into the liver cells into glycogen. That glucose then has now been removed from the blood and stored as insoluble glycogen, and therefore the blood glucose levels have been restored to normal. Now, if you haven't eaten food containing carbohydrates or you've done a lot of exercise, your blood glucose concentration will decrease. This is detected by the alpha cells in the islets of Langerhans in the pancreas. And it will cause the alpha cells to release glucagon, and also the adrenal glands will be releasing adrenaline. The second messenger model occurs to activate enzymes to hydrolyze the glycogen into glucose. That glucose is released back into the blood, and it brings those blood glucose levels back to the normal range. So we're going to have a look at some of those elements in more detail. So let's have a look at the action of insulin. First of all, it's the beta cells in the islets of Langerhans that detect when blood glucose levels are too high outside of the normal limits. And this then results in the beta cells secreting insulin in response to this. And these are the three key effects that insulin will have to decrease the amount of glucose in the blood. First of all, when that insulin binds to receptors on the surface of target cells, so on the cell surface membrane of the cells in the liver and also in the muscles, it causes changes to the tertiary structure of the channel proteins, which are the channel proteins responsible for transporting glucose. And this change in structure of them results in more glucose being absorbed from the blood into the cells by facilitated diffusion. So that's one way that the blood glucose levels will decrease. Not only that, but when the insulin binds to those receptors on the cell surface membrane of the liver cells, it causes more protein channels to be incorporated into the cell surface membrane. So both points 1 and 2 result in more glucose being absorbed from the blood into the cells. The final step is converting that glucose that is now in the cells instead of the blood into glycogen. And this step has to happen because if we don't convert glucose to glycogen, glucose is soluble, so it will lower the water potential of the cytoplasm in the cells, resulting in more water entering the cell by osmosis, and the cell might burst, or you might have lysis. So this conversion of glucose to insoluble glycogen is glycogenesis, and when the insulin binds, it also causes that to happen in the liver cells. So this is just a visual representation of some of those stages that we talked about. So we've got here the insulin hormone is bound to the complementary shaped insulin receptor on the cell surface membranes of liver cells. When it binds, what happens is actually a cascade of reactions that you don't need to know about, but it causes this release of chemicals, and those chemicals cause vesicles that are within the liver cells, which is what we're looking at here, these vesicles, which is just a sphere of phospholipid, and we've got the channel proteins embedded within that. And when these chemicals are released, it causes these vesicles, which contain those glucose protein channels, to move towards the cell surface membrane, and they then with that cell surface membrane. And as a result, you would then have all of these extra protein channels embedded within the cell surface membrane. So there's a larger surface area for that facilitated diffusion of glucose from the blood into the liver cell. The action of glucagon, again, there's 3 different actions. It will attach to the receptors on the target cells, and that would be the liver cells or the muscle cells. And when it binds, it causes the protein to be activated into adenylate cyclase. And this causes ATP to be converted into cyclic or cyclic AMP. That activates another enzyme, protein kinase, and that can then hydrolyze glycogen into glucose. So that is your second messenger model, how you have this 2 stages of activation of enzymes until we eventually get the hydrolysis of glycogen. It will also activate other enzymes to convert glycerol from lipids and amino acids from proteins into glucose. So this is the second messenger model. The glucagon binds to the glucagon receptors on the target cell, so for example the liver. That, once it is bound, causes this protein to change shape. And that change in shape to the enzyme adenyl cyclase or adenylate cyclase makes it activated. And the activated form can catalyze the reaction of converting ATP into cyclic AMP. And cyclic or cyclic AMP is the second messenger. So the cyclic AMP, which is this second messenger, is triggering the final stage to happen. It can convert the inactive protein kinase into an active form, and that is the enzyme which can then hydrolyze glycogen into glucose. Now, this second messenger model happens for glucagon binding, but also adrenaline. So it's exactly the same process when adrenaline binds. Now, adrenaline is the hormone that is secreted from the adrenal glands in response to anxiety, fear. So it's that flight or fight hormone. That's often how it's described. And the adrenaline will increase blood glucose levels. So if you did have to suddenly fight to survive or run away to survive, that would mean you'd need a lot of muscle contractions, which requires a lot of respiration, which requires glucose. So that's why adrenaline results in this increase in glucose, or that's why it's an advantage. The way it does that is, again, this second messenger model, much like we just saw for glucagon. So all of this is exactly the same as it was for the second messenger model for glucagon. It's just this time it would be adrenaline is the hormone that binds, and it binds to complementary shaped adrenaline receptors on the cell surface membrane of those liver cells, which are the target cells. But the cascades of responses resulting in the second messenger model is the same. The liver is involved in all 3 of these processes. Glycogenesis is when glucose is converted to glycogen, and this occurs in the liver and is catalyzed by the enzymes there. Glycogenolysis or glycogenolysis, pronouncing it like that so it's easier to remember, the lysis of glycogen is the splitting or hydrolysis. And again, this happens in the liver, and this is due to the enzymes involved in the second messenger model. Gluconeogenesis is creating glucose from other molecules, so the amino acids and glycerol, and again this happens in the liver due to the enzymes found there. Now, diabetes is when the body cannot produce enough insulin or it's not responding to insulin anymore. Type 1 diabetes is when people are unable to produce insulin or not producing enough. It normally starts in childhood and as a result of an autoimmune disease where the beta cells are attacked. So the treatment would be injections of insulin to counteract this. Type 2 diabetes, though, is when the receptors on the target cells lose their responsiveness to insulin. It usually develops in adults as a result of obesity and poor diet, and it's normally therefore controlled by regulating the intake of carbohydrates in your diet, increasing exercise, and sometimes it still involves insulin injections also. Osmoregulation is how the water potential of the blood is controlled. Blood with too low a water potential would be described as hypertonic. Blood with too high a water potential would be hypotonic. And if you had hypertonic blood, that means that too much water will leave the surrounding cells and move into the blood by osmosis, and that would cause your cells to shrivel or crenate. If you had hypertonic blood, that means that the excess water from the blood would move into the cells by osmosis, and it could actually cause the cells to burst. Now, neither of these are good, which is why we have to regulate the water potential. What could cause this is if you had hypertonic blood, that could be from excess sweating, not drinking enough, having a diet that's containing lots of ions, so salty diets. If you had hypotonic blood, then maybe you've drunk too much water or you don't have enough salt in your diet. So the corrective mechanisms for both. If you have hypertonic blood, more water has to be reabsorbed by osmosis into the blood from the tubules in the nephron, which is in the kidney. Therefore, you'll have urine which is lower in volume and more concentrated. If you have too much water in your blood, so hypertonic, that means less water will be reabsorbed by osmosis into the blood from the tubules in the nephrons in the kidneys. And as a result, you'll be producing larger volumes of urine, which are more dilute. Now, all of this happens in the nephron, and you need to know the structure of the nephron, which you find in the kidneys. The nephrons are these long tubules. They're surrounded by capillaries, and you have around 1 million of these in both of your kidneys. It's where the blood is filtered to remove the waste, but also selective reabsorption happens to reabsorb any useful substances back into the blood. So the way this happens is step 1, as the blood is going to flow into the glomerulus, the capillaries there, because there's a really, really high hydrostatic pressure, water and small molecules are forced out of the capillaries and into the renal capsule, which is this yellow part here, and the glomerulus is all of the red bit there where you have the capillaries. You've then created this filtrate, and that filtrate will then start to pass through to the proximal convoluted tubule. And this is where selective reabsorption occurs. So glucose is reabsorbed in the proximal convoluted tubule. The loop of Henle is where we maintain a sodium ion gradient, and that is to make sure that water can then be reabsorbed back into the blood. So at this point, the filtrate is going to be losing lots of the ions in the ascending limb, and that will then lower the water potential of the surrounding area around the loop of Henle, and then water will be able to move out by osmosis as it goes down the descending limb. So as the filtrate reaches the distal convoluted tubule, it's lost a lot of water, but also quite a lot of ions. And because it's lost a lot of ions, it is actually quite dilute by this stage. So more water will move out by osmosis, and at the collecting duct, even more water can move out to go back into the blood. The collecting duct carries the remaining liquid away to form the urine. Now that is a very basic overview. To have the full details, you will need to watch my full video where I actually go through every one of those steps in a lot more detail. This is just the basic summary video. That would not be enough detail though, so do check that one out up here. So how then this is regulated is using the hypothalamus and the posterior pituitary gland in the brain. The hypothalamus is where the changes in water potential are detected by receptors called osmoreceptors. If the water potential of the blood is too low, water leaves the osmoreceptor cells by osmosis, and that causes them to shrivel. And this stimulates the hypothalamus to produce more of a hormone called ADH, or antidiuretic hormone. If the water potential of the blood is too high, water enters the osmoreceptors by osmosis. This stimulates the hypothalamus to produce less antidiuretic hormone. So the hypothalamus is where the ADH is produced, but it then moves to the posterior pituitary, and from here it is released into the capillaries and into the blood. ADH travels through the blood and to its target organ, which is the kidney. Now, what that hormone does is it causes an increase in the permeability of the walls of the collecting duct and the distal convoluted tubule, so they're more permeable to water. And as a result, more water will move out of the nephron by osmosis and it's reabsorbed into the blood. So you'll have more water returning to the blood and more concentrated urine. So if we have a look then at this overall view of the homeostasis, negative feedback, we've got our normal water potential of the blood. Now the water potential of the blood could increase, so maybe you've drunk a lot of water in a short space of time. And that increase in the water potential of the blood gets detected by the osmoreceptors, which are within the hypothalamus in the brain. The hypothalamus will then produce less ADH, and therefore the posterior pituitary gland will release that less ADH into the blood. That transports around in the blood until it binds to the target cells, which are the cells lining the distal convoluted tubule and the collecting duct walls, and they then become less permeable to water. That means less water is reabsorbed into the blood and more stays in the nephron, which is then within the collecting duct, which goes on towards to make urine. So it results in a larger volume of dilute urine. And as a result, the water potential of the blood returns back to its normal limit. Now, the opposite of that would be if the water potential of the blood decreases, meaning you don't have enough water in the blood. And that could be you've not drunk enough water, or maybe you've been sweating a lot. And this again is detected by the osmoreceptors in the hypothalamus. And this time the hypothalamus will produce more ADH in response, which is then released into the blood by the posterior pituitary gland. That will then transport in the blood, bind to the receptors on the cells lining the distal convoluted tubule and the collecting duct, and that makes the walls more permeable to water. Therefore, more water is reabsorbed back into the blood, and as a result, there are lower or smaller volumes of urine, and it's more concentrated urine, and this results in the water potential of the blood returning into that normal limit. So the first thing with inheritance is there are a lot of key terms that come up quite often as a 1-mark definition just to start off an inheritance question. So make sure you know all of these definitions as they've all come up at some point in the past. So genotype is the genetic constitution of an organism. Phenotype is the expression of the genes, but also the interaction with the environment, so don't forget that bit. Homozygous is when you have a pair of homologous chromosomes carrying the same alleles for a single gene, whereas heterozygous is when you've got the homologous chromosomes carrying 2 different alleles for a single gene. Recessive allele is only expressed if there's no dominant allele present, whereas the dominant will always be expressed. Codominant is when both alleles are equally dominant and expressed in the phenotype. Multiple alleles is when you have more than 2 alleles for a single gene. Sex linkage is a gene whose locus is on the X chromosome. Genes are located on the same chromosome, not the sex chromosomes though, for autosomal linkage. Epistasis is when one gene modifies or masks the expression of a different gene at a locus. And then monohybrid and dihybrid is referring to types of genetic crosses. So monohybrid is when you're looking at the inheritance of just one gene. Dihybrid is when you're looking at the inheritance of 2 genes at a time. And we're going to go through the types of inheritance in more detail and have a look at examples of genetic crosses. Now, a big part of the inheritance topic is being able to determine the outcome of genetic crosses. So to help you with that all in one place, I've got you this genetic coding table. So for the different types of inheritance, this is how you would represent it in a genetic cross. A monohybrid is what you would have done at GCSE. So you pick a letter to represent the gene, capital is the dominant allele, lowercase is the recessive. For codominant, because they're both dominant, you can't go for a capital and a lowercase. So instead, the base letter is representing the gene, and the superscript letter is representing the allele. So for example, blood group is IB for blood group B, IA for blood group A, but if you have this here, IBA, that would mean that it's someone that's blood group AB, because both dominant alleles will be expressed. Multiple alleles, again, you can't use the upper and lower case because you have more than two alleles, and you might have this linked with the idea of codominance as well, as we see in the example of blood groups. The allele that codes for blood group B is dominant, the allele that codes for blood group A is dominant, but there are multiple alleles because there is a third one for blood group O, but that is recessive. For sex linkage, you have to represent this by showing, first of all, the X and Y chromosome. And in sex linkage, the gene is only found on the X chromosome. So you'll only be drawing the allele on the X chromosome, not on the Y chromosome. But we still stick to capital for dominant, lowercase for recessive. Autosomal linkage is always linked to dihybrid because it's looking at the inheritance of two genes found on the same chromosome. So we can still use the capital and lowercase, but we'd have two different letters representing the two different genes. Epistasis would be the same idea as well, because they often don't link it to any of the other types of inheritance because epistasis is complicated enough. So now for the examples. So we're starting off with a simple monohybrid example where we're looking at the inheritance of cystic fibrosis, which is caused by a recessive allele. And we're told in this question that we have two parents that are heterozygous, so we can see that represented here. And we're being asked what is the probability they will have a girl with cystic fibrosis. Now don't be thrown, this is not a sex-linked gene, but they still want you to work out the probability of, first of all, whether the child will have cystic fibrosis. And we can see here we have 25% probability that a child would have cystic fibrosis. But because they want you to say the probability of a girl having cystic fibrosis, there's 50% probability they'd have a girl. So we'd then do 25% times 50%, or half of it basically. So there's 12.5% probability that they'll have a child who has cystic fibrosis who is also a girl. Now if we have a look at a codominant example, cows can be red, white or roan in colour, and this is roan here where it's blotchy with both colours expressed. And in this example we have two roan cows reproducing, and they want to know what's the probability they'll produce red offspring. So two roan cows, because this is codominance, both the red and the white are dominant. That's how it's possible to get roan where they're both expressed. So here is how we'd represent it for the two parents. And then if we do our Punnett square, we can then see we'd have one roan, one red, actually a second roan down here, and one white. So that would mean we have 25% probability of getting a red cow from those parents. Looking at the inheritance of blood groups is an example of multiple alleles and codominance. So here we have the phenotypes that are possible of the different blood groups. Now you don't need to know this. If they wanted you to work out anything to do with blood groups, they would have to give you some information so you could work it out. It's not on the spec, you have to know the blood groups. It's just a really common example to test your understanding of codominance and multiple alleles. So blood group A, you could either have two copies of the dominant A or one copy of the dominant A and one recessive O allele. Same idea for blood group B, but with the B alleles. To be blood group AB, this is our codominance where we have one copy of both of those dominant alleles, so both are expressed. Blood group O is recessive, so it's only possible if you have 2 copies of IO, which is our recessive allele. So if we have a look at an example, we have parents with blood group AB and O reproduce, and we're told that A and B are codominant, O is recessive, because as I said, you're not expected to know that. It would be application of your knowledge. And they want to know what's the probability they'll have an offspring with blood group A. So the first thing is you have to state the parental genotypes. Then we can work out the gametes, which I've put here on the outside of the Punnett square. Then we have the offspring genotype and the offspring phenotype. So we can see the probability of having blood group A is 50%, because we have 2 individuals down here that have both got a genotype that codes for blood group A. So now our example of sex linkage, it's looking at colour blindness, which again you don't need to know, it's an application of your knowledge, and you're told that colour blindness is caused by a recessive allele found only on the X chromosome. If a non-colourblind male reproduces with a female carrier of the allele, what is the probability their children will be blind? So first of all, we need to work out the parents' genotypes. So the mother is a carrier, so that means they'll have one dominant allele and one of the recessive alleles that causes colour blindness. And then the male, XY, they do not have colour blindness, so that means they have to have just the dominant allele on their X chromosome. So here we have our gametes. We then have the offspring's genotypes and phenotypes. So the probability that they would have a child that is colourblind is 25%. So epistasis, we said, is when one gene influences or affects, masks the expression of another. So some examples are the coat colour in mice, or in Labradors, fruit colour of vegetables as well, such as squash. And we'll have a look at the Labrador example. So gene 1 controls whether pigment will or will not be expressed, and the dominant allele codes for pigment will be produced, whereas the recessive codes for no pigment, and therefore the dog won't be producing any of the darker pigments and it'll be that yellow colour. Gene 2 controls what colour the pigment will be if it is expressed. So dominant is black and recessive is brown. So that is how you get the 3 different colours in Labradors. So we have a look at an example. We're going to cross these 2 parents. So the first thing, we've been given the genotypes, but we have to work out the phenotypes. Now both of them have a dominant E, which means pigment can be produced, and they have a dominant B, which means they'll be black. After that, it's working out all the possible gametes that we could have. Then we'd need to do a Punnett square to work out all the possible genotypes of the offspring, and then the phenotypes of the offspring, which I've done for you just here. So we can see we end up with 4 yellow dogs because they have homozygous recessive for that E gene. So even though they might code for black and brown, we won't see that colour because it's been masked by the first gene. Now dihybrid crosses are when we have 2 genes considered at the same time in a Punnett square. So a common example is Mendel's peas. So we've got round and yellow, wrinkled and green as our 2 different traits, the shape versus the colour. And in all of the genetic crosses that we've gone through, it's these 6 features you have to have for all of the marks. We need the parental phenotype, genotype, the gametes, offspring genotype, and then you match the offspring phenotype to the genotypes, and finally give the proportions. So if we have a look at this example, with our round and yellow, round is dominant and yellow is dominant, wrinkled and green are recessive. And in this example, although we've got the 2 dominant traits, they are homozygous dominant. And they would have to tell you that in the question, or they would have to give you information so you could work it out. So then we'd have a look at our gametes, and each gamete should have 1 copy of each gene. So you need 1 of the letter R's for the shape, 1 of the letter Y's for the colour. Now in this instance, that would mean that all of the offspring would be heterozygous, and you'll always see this pattern. When a homozygous dominant parent is reproduced with a homozygous recessive parent, you will always get 100% of the offspring being heterozygous, and therefore showing the dominant trait for both of those genes. Now if we cross the F1 generation though, where both parents are heterozygous, there are now 4 possible different gametes for both parents. And this would be where we get these large Punnett squares, where we put every possible gamete for both parents in the outside box. Then we work out the offspring genotypes, and then we work out all of those phenotypes, and I tend to put it in the box, so you are then definitely getting that mark of matching the phenotype to the genotype. So our ratio here is 9:3:3:1. And if you have a dihybrid cross where both parents are heterozygous for both genes, then it will always be 9:3:3:1, unless there is autosomal linkage or there is crossing over. So let's have a look then at crossing over and how this could affect the ratio. So crossing over results in new combinations of alleles in the gametes. So that means the predicted gametes that we had in our Punnett square may actually differ. So autosomal linkage is when 2 genes are located on the same chromosome, but it won't be the X or Y chromosome. So we can see here we have gene 1 is found on the same chromosome as gene 2. And this is showing us the homologous pair of that particular chromosome. So what this means is that the alleles for each gene are linked on the same chromosome. So they have to be inherited together, because in meiosis, when gametes are made, that whole chromosome will be pulled to create one particular gamete, and this chromosome will then be pulled to the other pole to create the other gametes. So that would mean you would have to have the 2 dominant alleles for the Y and R gene inherited together, and the 2 recessive alleles for the Y and R gene inherited together, as those would be the only 2 types of gametes that could be made. So if we go back to the dihybrid example where we said we have 2 parents that are both heterozygous, we said these were all the possible gametes that could be made. But if there was autosomal linkage, even though we have 2 heterozygous parents, because the R and Y gene are linked on the same chromosome, and we can see we have the 2 dominants and the 2 recessive alleles on individual chromosomes, that actually means the only possible gamete would be one that has the dominant R and Y and one that has the recessive R and Y. So our possible genotypes of the offspring and the phenotypes are here, and we get a very different ratio, a ratio of 3:1, and that then does not fit the typical 9:3:3:1 ratio. So that is how autosomal linkage could prevent you from getting that ratio. But we also need to consider crossing over and autosomal linkage impact as well. So we've just talked through this example where, because of the autosomal linkage, we have a 3:1 ratio, but you could actually have this scenario here. Scientists actually observed the following results. So we have 4 different phenotypes, not just 2. And we can see we mainly had yellow and round, and the least common was the green and round. And this is only possible because crossing over has occurred to create new combinations of gametes. So not just these 2 gametes that we said would be possible, new combinations have been made, so there's now actually more gametes. So this is how it would have happened. We can see that we said we'd have the 2 recessive alleles linked and the 2 dominant alleles linked. If we then have crossing over, we can see that the recessive R and the dominant R cross over, that bit of the chromatid will break and exchange, and we now have 4 possible gametes instead. Now you also need to be able to see how chi-squared, the statistic, can link to inheritance. So chi-squared is used when you're investigating differences, but it's when your data is based on categories and frequencies. So if we go back to the very first example we had in this video, this is what we were looking at, the probability that 2 carriers would have a girl with cystic fibrosis. And we said we would expect the probability to be 12.5%. Now, chi-squared is the statistic used to investigate the differences between frequencies, but more specifically to determine whether there is a significant difference between the frequency we expect from our Punnett square and the frequency you actually observe. So let's go through an example. Corn can be purple or yellow and smooth or wrinkled. It's not actually just yellow like you see in the supermarkets, and this is determined by genetics. So using a Punnett square and by counting the actual number of each type of kernel, we can then use chi-squared to see, is there a difference between the expected frequency and the observed frequency? So does the frequency of the purple and yellow corn match the Mendelian genetics you would expect, which basically means the expected value you get from a Punnett square. So purple is dominant, yellow is recessive. And we've got this particular cross from two parents, and we would expect from this that we would get a 3-to-1 ratio of purple to yellow. A student then counted the actual number of each coloured kernel, and they recorded 21 purple and 13 yellow. So we can then use the chi-squared to see, do we have a difference between the expected ratio and the observed ratio? So we always state that there is no significant difference between the expected and the observed frequencies in the null hypothesis. And what we'd first have to do is convert the ratio, which is what we use in the Punnett square, into an actual expected frequency. So this is what was observed. So the total number of corn was 34. So if we have a 3-to-1 ratio is what we expect, the frequency that we would expect from that value would be 25.5 and 8.5. We would then do our observed minus expected. We square that, divide that by the expected, and we get our chi-squared value. In this example, the number of degrees of freedom is 1 because it's the number of categories minus 1. And we have 2 categories, purple or yellow. So the last thing you would need to do would be compare your chi-squared value to a critical value. And in this case, the calculated value of chi-squared we said was 3.176, and our critical value, we have 1 degree of freedom, so we need to look across at the row 1 and we go to 0.05, and that would be telling us that there'd be 5% probability, the results are due to chance. We always compare at this point to be able to say if we have a significant difference or not. And in this case, we can see that our calculated is less than the critical value. So that means there's more than 5% probability the results or the difference between the observed and the expected is due to chance. So that means we have to accept the null hypothesis, which was that there will be no difference between the expected and the observed frequency. And what that means is the expected frequency that we calculated using our Punnett square was the same as the frequency we observed. Or in other words, it did follow the Mendelian genetics that we expected. So you also need to know about the Hardy-Weinberg principle, which is essentially a mathematical model which can be used to predict the allele frequencies within a population, which means how common one particular allele is within that population's gene pool. So just to review some of these key terms then, gene pool, population and allele frequency. So the gene pool is all the alleles of all the genes within a population at one time. Population is all the individuals of one species in one area at one time, and they can interbreed to make fertile offspring. And lastly, the allele frequency, that's the proportion of an allele within the gene pool. So these are key definitions to learn for AQA, but it's also to help get your head around the calculation which we're going to go on to next. Now the Hardy-Weinberg equation is this one here at the top, but you actually use both of these equations simultaneously. And the main thing you need to be confident with is, first of all, knowing those equations, but secondly, knowing what each component means. So a single letter by itself is an allele, and the P is representing the dominant allele. The Q is the recessive allele. These ones here in blue are the genotypes. So P squared is homozygous dominant genotype, meaning 2 copies of the dominant allele. 2pq would be heterozygous genotype because we've got 1 of each letter, and q squared is the frequency of the homozygous recessive genotype. So we use these 2 equations to be able to work out any one of those components using the data you're given. So in this example, we're told that cystic fibrosis is caused by a recessive allele. So that would be the letter Q. 0.02% of the population suffer from cystic fibrosis. And you then need to use that information to work out what proportion are carriers. Now, the first thing you should always do is identify which parts of the formula you have values for. So if we've been told that that percentage of the population are sufferers, that means that is our genotype Q squared, because they must have 2 copies of the recessive allele to be sufferers. So we've been told that Q squared is 0.02%, but we can't leave it as a percentage because in our Hardy-Weinberg equation, we say that the total equals 1, not 100. So it's not as a percentage, we need it as a decimal place. So we divide that by 100 to turn it into a decimal. Next then, we work out P and Q. So if we have Q squared, that means we can work out Q by square rooting that value. So there is our Q value. And if you have Q, we can then rearrange that second equation, making P the subject, and it would be 1 minus Q. So P is 0.986. And once you know what P and Q are, you can work out any other part of the equation. Now, they've said they want you to work out what proportion are carriers. And if they're carriers, that must mean that they are heterozygous in this example. So they're asking you to work out 2pq. So 2pq would be 2 times p, so 2 times 0.86, times q, which is 0.014, so 2pq is 0.03. And you need to look carefully to see if they specify your answer has to be given as a percentage or not. If it doesn't say anything, then you can leave it as 0.03. If it says a percentage, then times it by 100. So next we're considering variation. And individuals within a population of the same species may show a wide range in variation of phenotype. And this is due to both genetic differences but also environmental factors. The main source of genetic variation is mutations, but there will also be some genetic variation introduced by random fertilization of gametes and also in meiosis, crossing over and independent segregation. Now that variation is what leads to natural selection. Predation, disease, competition are all things that are going to be driving this natural selection. And as a result, those organisms with the phenotypes providing the selective advantages are more likely to survive and therefore pass on their favourable alleles to the next generation. This changes the allele frequencies within the gene pool, because whichever allele gave the selective advantage will become more frequent. So that takes us on to this idea of types of selection. Now, in topic 4, you learn about stabilizing and directional. What we're going to be covering is just disruptive, because that's the only one that comes up in topic 7. And disruptive selection, which we can see up here, is when individuals which contain the alleles coding for the extreme traits are more likely to survive and pass on their alleles. So as a result, the allele frequency changes over many generations, and more individuals end up having those 2 extreme traits. And the middling trait is lost because having that trait is not an advantage to survival. So it won't be passed on to the next generations. Now that disruptive selection ultimately can result in speciation, which is the creation of a new species. And this occurs when one original population of the same species becomes reproductively isolated. And that isolation means that there are now 2 populations of the same species, but they will not breed together. So that can result in the accumulation of differences in their gene pools to the extent that those 2 populations would be unable to interbreed and make fertile offspring if they were then able to reproduce. So there has to be something that is preventing them from reproducing with individuals from those separated populations, and therefore over many, many generations of those 2 populations accumulating different mutations, we end up with them having such different DNA that they cannot make fertile offspring. Now that could be allopatric speciation, or sympatric. So allopatric is when there is a geographical barrier, so that could be a new mountain range, for example, whereas sympatric is when there are reproductive mechanisms preventing it. So in allopatric speciation, a population can become geographically isolated over time. It could be a new mountain range or new bodies of water separating the land masses, and that separates the original population into two. So they are now unable to reproduce due to that geographical barrier. Both of those populations will be accumulating different beneficial mutations, and over many, many generations, that will then result in their DNA becoming so different that they cannot interbreed to create fertile offspring. And therefore we class them as two new species. Sympatric, we said, is when the populations become reproductively isolated, and this is due to differences in their behaviour. So they are not geographically isolated, they are in the same area, but they still won't reproduce. This could be because a random mutation has impacted the reproductive behaviour. So for example, it might cause individuals to perform a different courtship ritual, or maybe they now become fertile at different times of the year. So even if they did mate, they wouldn't become pregnant, and therefore they would not be mixing genes within that particular gene pool. Due to this, these individuals will not produce together and there'll be no gene flow between the populations. Over time, these reproductively isolated populations, just like in the other example, accumulate different mutations to the extent that their DNA is so different they are no longer classed as the same species. Genetic drift is the change in the allele frequency within a population between generations, and there will always be genetic drift from one generation to the next, but continual, substantial genetic drift is what results in evolution. And the smaller a population is, the bigger the impacts allele frequency changes have proportionally, and this is why evolution occurs more rapidly in smaller populations. For this part of topic 7, the populations, there's a lot of key terms that can come up as just 1 or 2 mark questions in the exam. So these would be great to turn into flashcards or just to test yourself. You can screenshot and take a copy of this table. So populations is a 2 mark definition, and your definition is it's a group of organisms of the same species living in the same area at the same time. That first part is 1 mark. The second mark is that they can interbreed to make fertile offspring. Habitat would just be 1 mark, so it's the part of an ecosystem in which particular organisms live. Community, so this is all populations or all the populations of different species in the same area at the same time. And it is essentially the same, say, in the same area at the same time. The ecosystem, so that is the community and the non-living components of the environment, so essentially the biotic and the abiotic factors. And you need to have an awareness that the ecosystems can range in size from being very small to very large, like global. Niche is often a 2 mark definition as well. So it's an organism's role within an ecosystem, including its position in the food web, but also its habitat. And it's normally split as it's an organism's role within an ecosystem, and then the second mark is also the habitat also. And each species occupies its own niche, and no 2 species can occupy the same. And if they do try, then that's when you get competition, and one species will eventually be outcompeted. Carrying capacity is the maximum population size that an ecosystem can support. And if you exceed that carrying capacity, there then won't be enough resources for all of the individuals and they'll start to die, and that will then bring it back to the carrying capacity level. Abiotic factors are the non-living conditions of an ecosystem that affect the survival of individuals. And the biotic factors are the impacts of the interactions between organisms. So if we have a look then at the abiotic factors and the biotic factors. Abiotic factors, as we said, are the non-living components of the ecosystem, and these can be factors like the temperature, oxygen and carbon dioxide concentration, light intensity, pH and soil conditions. Plants and animals are adapted to the abiotic factors within their ecosystem. These adaptations develop through the process of natural selection over many generations. The less harsh the abiotic factors, so for example if there's plenty of water and light, the larger the range of species and the larger the population size. So for example, in the desert, because it's very harsh conditions, you'll have a smaller range of species and the population size is smaller. Compared to down here where we can see there's plenty of light and water, we have a bigger range of species and there's more of each species. Biotic factors, we said, are the living components that are interacting together. So for example, we could be looking at competition, whether it's interspecific or intraspecific, and also the effect of predation. So our two types of competition. Interspecific competition is when members of different species are competing for the same resources that are in limited supply. Whereas intraspecific competition is when members of the same species are competing for the same resources and also a mate. And competition for a mate links to the courtship rituals. The predator-prey relationships you will see presented in graphs like this, which are called predator-prey graphs. And regardless of the species, the graph always follows a very similar pattern. The size of the predator and prey populations will both fluctuate. There will typically be more prey than predators because there has to be more prey to sustain the predators because they are eating them. And the size of the population change will always have a slight lag. So whatever happens to the prey, that will then happen to the predators shortly after. So the prey will increase first, then the predators will. The prey will then decrease because they're being eaten by the predators. But then because their population decreases, so will the predators. So if we think about sampling, first of all, why do we have to sample? Well, if you were asked to work out how many daisies there were in this field, you would have to do sampling rather than count every individual one. It's more time efficient and you're actually more likely to get an accurate answer because you'd probably miscount if you had to count all of those. But to make sure sampling is actually going to accurately represent that population size, you have to make sure that you are randomly sampling if it is a uniform area, and that's to eliminate bias. You also should have a large sample, so take at least 30 samples. Now I've got here a sampling summary just to help you see how it all links together. So if you have a slow moving or non-motile organism like plants, you would sample using a quadrat. If it was uniform distribution, you would do random sampling. If it wasn't evenly distributed, so for example a river in the middle of your field, then you would do a line transect. If you have a motile organism, meaning they can move, then you would need to use mark, release, recapture. So let's have a look at these three types of sampling. So first of all, random sampling. This is when you'd have two tape measures pulled out at right angles to create a gridded area. You would then need to use a random number generator to get two coordinates, and those coordinates indicate where you would place your quadrats. Once you place your quadrat, you then collect the data, and you would then repeat this at least 30 times and calculate a mean. Now, if you didn't have an even distribution, we would use a line transect. So common examples of when you need a line transect are rocky shores or any kind of beach fronts, as well as maybe going across a path or a river. So for example, if we wanted to have a look at the impact of the river, you would put your line transect going across it. If you wanted to know the impact of the rocky shore, you'd go from the rocky shore going further away into the land. You can either do a belt transect or an interrupted belt transect. A belt transect is when the quadrat is placed at every single position along the tape measure. An interrupted belt transect is when you place it every set distance. So it might be every 2 meters, every 5 meters, whatever seems appropriate for the area you are sampling. So for this method, you would place the tape measure at a right angle to the shoreline, just like we see here. You would then place your quadrat every 5 meters, or whatever position distance you're going to state. Collect the data that you can see in the quadrat, but you still have to repeat this multiple times. But you would be repeating this by placing another 30 transects along the beach at right angles to the shoreline. And you have to be really specific with how you word number 4. If you just said repeat 30 times, you won't get the mark. You have to describe the position. Now, mark, release, recapture you would use for moving animals. So first of all, we have the method here. An initial sample of the population in question is captured. These individuals are then marked and the number caught is recorded. And the mark has to be all weather resistant so it doesn't come off, but it also has to cause no harm to the organism. The marked individuals are then released, but you have to leave them for long enough to evenly distribute across their habitats before you then take your second sample. And when you take your second sample, the total number captured in that sample is recorded, as well as how many you recaptured that had the marking on them. And the size of the population can then be calculated using that information. But as with all sampling, the more times you repeat it, the more reliable your results should be. So here's our calculation. To estimate the total population, you would do the number of organisms initially caught times by the number of organisms in the second sample, divide that by the number of organisms that you recaptured. So here we have an example where we caught 20 butterflies. In the second sample we caught 22 and 9 of them were from the original 20 that were marked. So that would mean we had 48.89, but we have to round up to have a whole number. So 49 butterflies. Now you have to consider the ethics here of how you capture them. And you aren't meant to be an expert and know exact suggestions. What you need to know is how you capture them and how you mark them must cause no permanent damage. So don't use anything that's going to be toxic, and it can't increase the chances of predation, and it mustn't interfere with their reproduction either. There are some assumptions with this as well. So the estimate may not actually be very accurate because of these assumptions, because we assume that the population size is constant, meaning there's no birth and death and no migration, which is very unrealistic. We also assume that the animals do redistribute evenly, but that might not be true. They might all huddle together near a food source. So next we have a look at succession, which is the change in an ecological community over time. A primary succession starts with a pioneer species colonising bare rock or sand. And the pioneer species, such as lichen, are adapted to survive in harsh abiotic factors. And through their death and decomposition, they change the abiotic factors to become less harsh, and this forms a thin layer of soil, which we call humus. Mosses and smaller plants are now able to survive in that area, and they further increase the depth and nutrient content of the soil. And this pattern continues, and as the abiotic factors continue to be less harsh, larger plants can survive and they continue to change the environment further. Each new species may change the environment in such a way that it becomes less suitable for the previous species, and therefore those previous species become outcompeted. Changes that organisms produce in the abiotic environment can result in a less hostile environment and increases the biodiversity. The final stage of a succession is known as the climax community, and this is dominated by trees typically. Now, a secondary succession is when the succession is disrupted and the plants are destroyed. Therefore, the succession has to start again, but we don't start again from bare rock because the soil is already there, it's just the plants that have been destroyed. So overall, from succession, as we have that change over time, the species richness will increase, as well as the number of organisms, or in other words, the biodiversity increases over time. As succession occurs, larger plant species and animals will also start to colonise the area. Now, often conservation of habitats involves controlling succession. The destruction of habitats, usually due to human activity, results in a loss of food and a loss of space or habitat for organisms, and it can lead to extinction. So that is why we conserve the habitats, and succession is often managed to do this. So by maintaining an earlier stage in succession and preventing the climax community, a greater variety of habitats are conserved, and therefore we get a greater range of species. There needs to be management though between the conflicts between human needs, such as needing the wood for timber, and conservation in order to maintain the sustainability of the resources. So a compromise could be that forests are coppiced to provide timber for fuel, while still allowing the trees to survive. Now this picture isn't actually showing coppicing, this is showing that all the trees have been cut down. Copacine is when you cut down just part of the tree so they can still then grow. So we begin with mutations, specifically gene mutations, which are a change in the DNA base sequence of a gene. And this mainly occurs during DNA replication, which is within interphase of the cell cycle. They occur randomly and spontaneously, but the exposure to mutagenic agents, so things like ionizing radiation, do increase the frequency at which they can occur. If a gene mutation does occur, then it could result in a different amino acid being coded for, and therefore a different sequence of amino acids in the primary structure of a protein. This could cause the hydrogen and ionic bonds in the tertiary structure to form in different locations, and therefore it results in a different tertiary structure 3D shape. And it's the shape of a protein that determines its function. So that is why a mutation could result in a different function or a non-functioning protein. Now, if that mutation happens in a gene that controls the cell cycle, it could then result in cancer. So there's 6 types of gene mutations that you need to know about, and we'll go through each of these 6 just to demonstrate what they actually mean. So first of all, if we have a look at addition mutations, this is when one extra nucleotide and therefore base is added to the sequence. So here we have our original example of a DNA sequence, and we've added in an extra base. Now the result of that we can see is that all of the subsequent bases are shifted along one position, and therefore after the mutation all of the codons are changed. And we also have one extra base just there that won't even code for a whole amino acid. So it's very likely that this will code for a completely different tertiary structure, and therefore either a non-functioning protein or a protein with a very, very different function. Deletion's a similar idea. So you delete a base this time and you still get what's called the frame shift, but all of the bases after the mutation shift back one position. So every single codon after the mutation is changed, and also you'll have one fewer codons because we now only have 2 bases in this final codon because one of the earlier bases was deleted. Substitution is where one of the bases is swapped for another one. So again, we can see we've got our original sequence, and here's the new sequence where we've had a mutation occur, so thymine has been swapped for adenine. Now this could result in a different amino acid being coded for, or it might actually result in the same amino acid being coded for because the genetic code is degenerate. It could also have no impact on the protein being coded for if this substitution happens within the introns, because introns do not code for amino acids. An inversion mutation is when a section of the bases detaches from the DNA sequence, and when it rejoins, they are the other way round, so they're inverted. And that's what we can see here. This section, TTC-AGG, that has been removed, but then when it was replaced, it's back to front, it's inverted. So that particular section of the DNA will now probably code for different amino acids, and therefore you'll have a very different primary structure. Duplication is when one particular base is duplicated at least once in the sequence. So again, you'll have a frame shift just like with the addition, but it's going to have even more of an impact because you're adding in even more bases. Translocation is when a section of bases on one chromosome detaches and attaches onto a different chromosome altogether. Now this is a substantial alteration and can cause significant impacts on gene expression, and it can cause therefore a very, very different phenotype. Now multicellular organisms have a range of specialised cells, and those all originated from stem cells. And stem cells are undifferentiated cells that have the ability to continually divide and also become specialised. There are different types of stem cells as well, and this is determined by the differentiation abilities that they have. So you can have totipotent, pluripotent, multipotent and unipotent cells. So if we have a look at the totipotent first, these stem cells can divide and produce any type of body cell. So they have the ability to differentiate into any cell. And the totipotent cells occur only for a very, very limited time in the early mammalian embryos. Pluripotent stem cells are also found in embryos, and they can divide into almost every cell, but not the placenta cells. So they can divide an unlimited number of times as well, and they're used in treating human disorders. The multipotent and unipotent stem cells are found in mature mammals, so adult cells, and can divide to form only a limited number of different cells. Unipotent can only become one type of cell. Multipotent is a very limited number. So the bone marrow is multipotent stem cells. It can only make blood cells, for example. And unipotent stem cells, an example, is making cardiomyocytes. Now, there are quite a few ethical issues with using pluripotent stem cells from embryos, and that is why induced pluripotent stem cells are now used. So these can be produced from adult body cells, that's what somatic means, using protein transcription factors to overcome the ethical issues. So what you would do is take a sample of the adult cells, which we can see here, a sample from the liver, and if you switch the genes off that made those cells specialized, then you can turn those cells back into their pluripotent state. So this links to the theory of transcription factors. And that leads us into transcription factors. So this is how transcription is controlled. We'll be looking at how translation is controlled later. So in eukaryotes, the transcription of target genes is stimulated or inhibited when specific transcriptional factors are moving from the cytoplasm into the nucleus. And when they do that, they can bind to receptors into the DNA to either turn on or off genes. And what that means is that gene will either be transcribed or it won't, and therefore the protein will either be coded for and created or not. And it's this turning on and off genes which makes cells specialized. So how this works then? Transcription of a gene only occurs when a molecule from the cytoplasm enters the nucleus because it has to bind to the DNA, and the DNA is only found in the nucleus. These molecules are proteins, and each one can bind to a different base sequence on DNA, and that will be how it initiates the transcription of a gene. Once bound, that is when transcription can then begin, and mRNA is created. That is then used in translation to create the polypeptide chain. So if the transcription factor does not bind, the gene is inactive, meaning transcription won't occur and the protein won't be made. So what we can see here is a transcription factor. So we have our DNA binding site. So this is the part that will bind onto the DNA. And we have a receptor here because often another molecule has to bind to the transcription factor before it can work. And that is where estrogen comes in. Estrogen is a steroid hormone that can initiate transcription. The way it does this is, first of all, because it's steroid based, which is a lipid, it can diffuse through the cell surface membrane into a cell. It will then bind to the receptor. So estrogen is complementary in shape to the binding site of the receptor on the transcription factor. When it binds, it causes the DNA binding site to slightly change. That transcription factor will then move into the nucleus, and because the DNA binding site has changed shape, it's now complementary to the DNA so it can attach and initiate transcription. mRNA is then created, which can then leave the nucleus and be used in translation. Now, another way that protein synthesis is controlled is through something called epigenetics. And epigenetics is the heritable change in a gene function, but the DNA base sequence itself is not changed at all. Now, these changes can be caused by the environment, so things like your diet, stress levels, exposure to certain chemicals, and this can inhibit transcription. So one type of impact is increased methylation of DNA. Now what this means is if you have lots of this chemical tag, methyl groups, binding to the DNA, it actually inhibits transcription. And the reason for that is when the methyl groups are added to DNA, they attach to the cytosine base and it prevents transcription factors from binding. It will attract the proteins that condense the DNA histone complex, and in this way it prevents sections of DNA from being transcribed. So that's what we can see happening here. The methyl groups are added, and because they are an opposite charge to the DNA, which is slightly negative in charge, it causes it to attract and to coil and bind up more tightly. And because that DNA is coiled up more tightly, there is no space for the transcription factors to be able to bind onto. And that is how it will inhibit transcription. Now the opposite is acetylation, and that is when acetyl groups are added. But they don't bind to DNA, they actually bind to the histone protein, which is what the DNA wraps around. And decreased acetylation, so if you don't have very many acetyl groups bound to the histones, that will also inhibit transcription. If acetyl groups are removed from the DNA, then the histones become more positive. Now just to emphasize, where it says here from the DNA, it does mean it's still bound to the histone groups, but they're within that DNA complex. But if you take away the acetyl groups, which are negative in charge, that means the histones themselves are more positive, and they'll then be able to attract the phosphate group on the DNA more, which is negative. And that makes the DNA and the histones more strongly attracted and associated, and again it coils up more tightly, preventing the transcription factors from being able to bind. So decreased, if you don't have very many acetyl groups bound, you also have transcription being inhibited. So in summary, if we think about just the methyl groups, if you don't have very many methyl groups bound, then that means the DNA is less tightly coiled, so it can be transcribed. And we call it euchromatin at that stage when it isn't very tightly coiled. If you had lots of methyl groups bound, so increased methylation of DNA, that results in the DNA and the histones tightly coiling up and the transcription factors can't bind, so transcription is inhibited. If you have increased acetylation of associated histone proteins, again, that would result in the less tightly coiled active euchromatin, so transcription can occur. But if you have decreased acetylation, you have heterochromatin, which is when all of the DNA and histones are tightly coiled, so the transcription factors cannot bind, and therefore transcription is inhibited. The final way that gene expression can be controlled is through RNA interference, or RNAi. And that happens at this stage here. So the DNA has been transcribed to make mRNA. The mRNA leaves the nucleus, but we're going to be looking at how translation can be controlled or interfered with using RNAi. So in eukaryotes and some prokaryotes as well, translation of the mRNA produced from target genes can be inhibited through RNAi. And this is when an mRNA molecule that's already been transcribed gets destroyed before it's translated, and therefore it won't create the polypeptide chain. And this is done using small interfering RNA, or siRNA. So this is the molecule that is going to destroy the mRNA, and that's why this is called RNA interference, because our mRNA has been interfered with. So let's have a look at how that happens. Transcription results in the production of mRNA, and mRNA is then used in translation by attaching to a ribosome, and that creates the primary structure of a protein, or the polypeptide chain. However, if there is small interfering RNA, that can disrupt the mRNA that's been created and destroy it, and therefore it prevents translation from occurring. And this is what we mean by RNA interference. So small interfering RNA results in this interference. So that's just a concept map just to show you how this all links together. Now, how this actually destroys the mRNA is you have some double-stranded RNA, but there's an enzyme that can cut that and make it single-stranded, but also into small pieces. So it's small and it's going to be interfering, so siRNA. That will then bind to an enzyme, and that enzyme's called RISC, but you don't need to know that's the name. Because it's single-stranded, this complex of the siRNA and the enzyme can bind to the mRNA molecule that was made in transcription. And that then means that the enzyme is able to cut up that mRNA into small pieces, and therefore it cannot be used in translation. It won't be able to bind to the ribosome, and because it's cut up, you can't translate that entire sequence. The next thing you need to know about is cancer, which we briefly mentioned at the start. So it can result from mutations in genes that regulate mitosis. And if these genes do mutate and a non-functioning protein is created, then that means mitosis cannot be controlled. And that is why you end up with this uncontrollable cell division, and that is when a mass of cells or a tumour is created. Now, there are different types of tumours. You can have what's called a benign tumour or a malignant tumour. And you do need to know the differences in the properties of these. So a benign tumour can grow very large still, but it will be dividing at a slower rate. So it will take longer for there to be a larger tumour. Benign is non-cancerous, and one of the reasons it's classed as non-cancerous is they produce this adhesive sticky molecule, and that will then surround it so it cannot move. It's also in a capsule as well, which means that these cells that are having this uncontrolled mitosis, they're not able to break off and spread to other parts of the body. And therefore, if surgery can be conducted on the part of the body that the tumour is located, you may be able to cut out and remove that entire encapsulated tumour, and then it'd be very rare that the tumour would regrow. So the impact is localised, and it's often non-life-threatening because you can cut it out and remove it. But it does, as I said, depend on the location of the tumour. So, for example, a benign brain tumour is still very dangerous because any type of brain surgery is very high risk because you have to cut through the skull, and in removing that tumour, you could actually damage some of the other brain tissue. Now, in contrast, malignant tumours, these are the ones that are classed as cancerous, and they do grow much quicker. The cell nucleus becomes very large and it returns back to its unspecialised state again. They don't produce that adhesive sticky layer or the capsule, and therefore they can metastasise. And what that means is cells of the tumour can break off, spread through the blood, and therefore lodge in other tissues in the body, and then you get secondary tumours in other organs. The other thing that we can see here is because it's not encapsulated, yes, it also links the idea of metastasis, but also it can grow projections and reach the blood supply. And once that tumour has its own blood supply, it's going to be receiving lots of oxygen and glucose for respiration, and that is one of the reasons why those cancerous tumours can grow so rapidly. It can be life-threatening, and the removal of the tumour needs often supplementary treatment like radiotherapy and chemotherapy, and the reoccurrence is more likely than with a benign tumour. So the tumour development then, as we said, it could be due to a gene mutation, and if it is a gene mutation, the mutation would have had to have occurred either in the tumour suppressor gene and or the oncogene. It could also be linked to epigenetics though. So if you have abnormal methylation of a tumour suppressor gene or an oncogene, or if you have increased estrogen concentrations affecting transcription. So those are all of the possible causes of these tumour developments. Now, we've used this term tumour suppressor gene and oncogene, but let's just go through what those genes code for. So oncogenes are the mutated version of a proto-oncogene. And proto-oncogenes code for proteins which are involved in initiating DNA replication in the cell cycle. An oncogene, though, can result in the process being permanently activated to make cells continually divide even if new cells aren't needed in that part of the body currently. Tumour suppressor genes code for proteins which are involved in controlling the cell cycle, and they're involved in slowing down the cell cycle or to cause cell death. Now, if a mutation results in the tumour suppressor gene, that means that protein isn't produced and cell division isn't slowed down, so it'd continue. Mutated cells would not be identified and destroyed. Now, we said that epigenetics can also link to the cause of tumours, and that was because of abnormal methylation. So we looked at how methylation can cause genes to be turned on or off, and if a tumour suppressor gene becomes hypermethylated, that would mean there's an increased number of methyl groups bound to it, and that results in the gene being inactive and it is switched off. So if that happened to a tumour suppressor gene, that means that protein won't be produced that slows down the cell division in the cell cycle. Now, the opposite could occur in an oncogene. So as they may be hypomethylated, meaning you don't have very many methyl groups attached, that could result in the gene being permanently switched on, and therefore lots of the protein is being produced to initiate that cell division constantly. Now, the final thing we said is estrogen also has an impact in increasing the risk of cancer, and it's linked to this specific example. So estrogen is produced by the ovaries to regulate the menstrual cycle. However, after the menopause, the ovaries stop producing estrogen, and instead the fat cells in the breast tissue produce estrogen instead. And this is what's been linked to breast cancer in women post-menopause, and that is why age is one of the risk factors of developing breast cancer. So we looked at earlier how estrogen is a molecule that can bind to receptors on transcription factors. So if that is going to happen to a transcription factor involved in the cell division, so for example, it's a transcription factor that can bind to the DNA of a proto-oncogene, that would mean that that particular gene is permanently turned on, activating cell division within the breast tissue. Now, it has a knock-on effect as well, because as the tumour grows, because those tumour cells are breast tissue cells, that will result in even more estrogen being produced, and therefore even more of those proto-oncogenes could be switched on, activating more cell division. So this is an example of positive feedback. The more that we have of the tumour, the more estrogen, but that results in even more tumour cells occurring. The next bit of the spec is looking at the genome, and the genome is the entire genetic material of an organism in the nucleus of a cell. And sequencing of this genome, what that means is identifying the exact DNA-based sequence for all of the DNA in a cell. And many organisms' genomes have now been sequenced by scientists, including the human genome, which took about 13 years, but it was finally completed in 2003. And that's the timeline we can see here. Now, sequencing methods being used to identify this DNA-based sequence are continuously being improved and now automated. And that's why AQA have taken it off the spec. You don't need to know any exact methods because they are so frequently improving that if you did learn one, it would probably be outdated within a year. Now, there have been methods such as the Sanger method, but as I said, you don't need to know that because the science of how this is done is so fast changing. All AQA want you to be aware of is that fact, that sequencing methods are now automated and they're continuously being improved. Now, simpler organisms like prokaryotic cells, You learn earlier on in the course that those DNA do not contain introns. And that means that the genome can be used to directly sequence the proteins that derive from the genetic code, or in other words, the proteome. And that's really useful for many reasons. For example, identifying potential antigens on a microorganism like a bacteria to then create an antigen for a vaccine. More complex organisms like humans and eukaryotic cells, other eukaryotic organisms, have introns. And we also have regulatory genes in our DNA. And due to this, the genome can't easily be used to translate to the proteome. And the proteome is the entire set of proteins within a particular cell. Now, the next bit of topic 8 is looking at gene technologies. For example, recombinant DNA technologies. And I've put all of the rest of topic 8 onto one flow diagram here, and we're going to go through it a section at a time. So the three key concepts are creating DNA fragments, the idea of genetic fingerprinting, and then the uses in genetic screening, counselling, and locating of genes. So gel electrophoresis is actually used in genetic screening, but also genetic fingerprinting. Genetic screening, you're screening for genes and personalised medicines. Genetic fingerprinting is used in forensic medicine, forensic science, medical diagnosis, plant and animal breeding and paternity tests. But what we're going to be focusing on first is this left-hand side. So creating DNA fragments, we're going to look at what are the three methods for how DNA fragments can be created, how those fragments can then be cloned either in vitro using PCR or in vivo looking at the use of sticky ends, plasmids and gene markers. So we're going to look at creating DNA fragments first. So recombinant DNA technology, what this means is you're recombining the DNA of two different species. And this is what enables scientists to manipulate and alter genes to improve industrial processes, but also medical treatments. Say, for example, manipulating the DNA in bacteria so it can produce human insulin. And the first step in these technologies is you have to isolate the fragment of DNA that you want to recombine or insert into another organism's DNA. And there's three methods: reverse transcription, restriction endonucleases, and the gene machine. So reverse transcription, as the name suggests, we are doing the opposite of transcription. So we're using an mRNA molecule to create a DNA copy. So the enzyme that is used is going to make copies of DNA from mRNA. And the enzyme is reverse transcriptase. And reverse transcriptase naturally occurs in viruses like HIV. What you would then do is you would find a cell that produces lots of the protein of interest. Then from that cell, you should be able to find large amounts of the mRNA that will code for that protein. You can then isolate that mRNA and add the enzyme reverse transcriptase. And that will join the DNA nucleotides with complementary bases to the mRNA sequence. You will then have created a DNA sequence, but it will only be single-stranded. To make the DNA fragment double-stranded, the enzyme DNA polymerase is then used to create that second strand. So this is what we can see here. We've got our mRNA molecule, which is single-stranded. You would then add the enzyme reverse transcriptase, and free-floating DNA nucleotides would align opposite their complementary base pairs, and we've then created a single strand of DNA. We'd then add DNA polymerase to make it double-stranded. And the advantage of this option is cDNA, which is what we call the molecule that's made, is intron-free. And that's because it was copied from mRNA, and mRNA already has the introns removed. The second option is using restriction endonucleases, and these are enzymes that cut DNA. They naturally occur in bacteria as a defense mechanism to cut up the DNA of any invading organisms. And there's many different types of restriction enzymes that have an active site complementary in shape to a particular DNA base sequence. And we call that the recognition sequence or recognition site. And therefore each enzyme will cut the DNA at a particular location. Some enzymes cut at the same location in the double strand, and that would result in a blunt end. Other enzymes cut to create staggered ends and therefore exposed bases, and we call those sticky ends. Now that's what the diagram is representing here. This enzyme, HIND3, cuts the DNA, but it doesn't cut straight down, creating two straight edges, which is what we call a blunt end. It cuts in this staggered way to create exposed bases on both of those cut chains, and we call it sticky because there's the potential for complementary bases to align and then they join together. Now the palindromic sequence, that is referring to where it cuts. The top part of our DNA sequence is the same as the bottom part backwards. So palindromic means it's the same forwards as it is backwards. The third option is the gene machine, and this is when DNA fragments are created using a computerized machine in the lab. So the first part of the process is you'd have to examine the protein that you want to create lots of, identify what the amino acid sequence is, from that work backwards to sequence what the mRNA would be, the mRNA sequence, and therefore the DNA sequence. You would then enter that DNA sequence into the computer. There'd be checks for biosafety and biosecurity, that the DNA being created is safe and ethical to produce. The computer can then create these small sections of overlapping single strands of nucleotides that make up the gene, and we call those oligonucleotides. The oligonucleotides are then joined to create the DNA for the entire gene. PCR, which we'll be looking at shortly, can be used to amplify the quantity that you have, and it makes it double-stranded. So this process is very quick, it's accurate, you can design it to be intron-free, so that means the DNA can be transcribed in prokaryotic cells. So that's creating our DNA fragments. The next thing is we're going to look at how those DNA fragments can be amplified or cloned. And the first option is in vivo, which means cloning these DNA fragments inside of a living organism. So here are our stages of in vivo cloning. We've already looked at creating the DNA fragment, so the next step is how that fragment is inserted into a vector and how the host cell will then incorporate that vector. So those are the two bits we're going to look at first. Now, restriction endonucleases are used in this particular method. So in vivo cloning, you have to create your DNA fragment using a restriction endonuclease. And we can see here that enzyme has cut the DNA at the recognition site, and we have these sticky ends. Now, the DNA fragments need to be modified before they are used to make sure that transcription definitely occurs. One modification is a promoter region is added, and this is added at the start of the DNA fragment, and a promoter region is a sequence of DNA bases, which is the binding site for DNA polymerase. So that is to make sure the enzyme can definitely attach and transcription occurs. The second modification is a terminator region is added, and this is put at the end of a gene, and it causes RNA polymerase to detach and therefore stop transcription, so only one gene at a time is copied. So then we need to see how is the DNA fragment inserted into a vector. But in case you're not sure what a vector is, first of all, a vector is something that will carry the DNA fragment into the host cell. So whatever organism it is that you want to now contain that DNA fragment, the vector will transport it into it. And often plasmids are the vectors that are used. And plasmids are just loops of DNA that are sometimes found in bacteria. So the way that we insert the DNA fragment into the plasmid, which is our vector, is we cut open the plasmid using the same restriction endonuclease that was used to cut the DNA fragment of interest. That means that the same sticky ends are going to be created on the plasmid as the sticky ends that you have on the DNA fragment. So then when you mix your DNA fragments and the plasmid together, you should have complementary base sequences opposite each other so that the two pieces of DNA can align, and then you add the enzyme DNA ligase to join those nucleotides together. And that's what we're just seeing here, that the enzyme DNA ligase is going to be joining those nucleotides together by catalyzing the condensation reaction to create those phosphodiester bonds. So we now have, hopefully, the DNA fragment inserted into the vector. The next step then is getting that vector into the host cell where you want the gene to be expressed. So to do this, the cell membrane of the host cell must be made more permeable so that we increase the likelihood of these plasmids being able to move into the cell. Now to do that, you can mix calcium ions with the host cell and heat shock the cell. And what that means is you have to have this sudden increase and then decrease in temperature. That affects the permeability, and therefore the vector is more likely to enter the host's cytoplasm. Now the final thing is, in this in vivo process, is how you can actually check to make sure that the plasmid definitely took up the DNA fragment, and how you can check that the host cell definitely took up that plasmid. And that is where these gene markers come in. And that's what we're going to look at next. How you can identify whether the cells were transformed, meaning they took up the modified plasmid. And then if you do have cells that have, the final step will be growing lots and lots of those transformed cells. But we're just going to focus on how to identify transformed cells. Now there's three issues that might have happened, which is why we have to check that we do have transformed cells that contain the recombinant plasmid. Now issue number one is that plasmid that you've created, which is our vector, maybe it didn't actually get inside of the host cell, which might be a bacteria cell. The second issue could be that maybe the plasmid, it had sticky ends, it might have just rejoined with itself and sealed back up. So you might have plasmids that did get in, but they don't contain the DNA fragment, so that's not going to be useful. The final thing that could happen is that DNA fragment that you created, it could actually loop around and join together on itself, creating a mini plasmid. And therefore it's not attached to the plasmid, it's not going to be able to enter into the cell, and it won't work. So that's why we have to try and examine and identify which cells do contain the plasmid with the DNA fragment inserted into it. So one method is using marker genes. And marker genes are genes that are occurring on the plasmid, and we use them to identify whether the host cell, which in this case we're thinking about bacteria, successfully took up the recombinant plasmid. And the three types of marker genes which are commonly used are genes for antibiotic resistance, genes that code for fluorescent proteins, and genes that code for certain enzymes. So we're going to go through the antibiotic resistant marker genes option first. So we have here a plasmid which contains two marker genes. One gene is one that makes the bacteria resistant to the antibiotic tetracycline, and the other one is a gene that produces a protein to make the bacteria resistant to the antibiotic ampicillin. And here is our DNA fragment. Now that DNA fragment is deliberately inserted in the middle of the tetracycline gene. And in that way, it disrupts the tetracycline gene, and the bacteria would therefore no longer be resistant to the antibiotic tetracycline. So if that bacteria was exposed to tetracycline, the bacteria would die. So we then grow the bacteria on agar, and we can see these different colonies have grown. We then transfer a copy of those colonies in the exact position using this velvet block. So you basically stamp it on top of your agar plate, and then you place that on top of a new agar plate. And this time it's an agar plate with the antibiotic ampicillin dissolved within it. And what we can see is which colonies now are killed off because they are not resistant to ampicillin. We then do the same thing again. Now we transfer those colonies onto a second plate which contains the antibiotic tetracycline within the agar. And we can see we now only have 3 colonies left. So we can analyse these 3 agar plates and the different colonies that are present to work out from these bacterial colonies which of those bacteria didn't take up a plasmid at all, or which took up the plasmid but it didn't have the DNA fragment inserted in it, or which took up the plasmid and it did have the DNA fragment inserted in it, because that is the one that we're interested in, because we want that DNA fragment inserted in. So we can see here in this first one, this first copy plate, that if there is ampicillin in this agar, that means only bacteria which contain the plasmid at all would be able to grow. Because if it didn't contain the plasmid, it wouldn't be resistant to ampicillin and the bacteria would die. So that means only these remaining colonies definitely contain a plasmid. But what we don't know is whether it's the original plasmid or the plasmid of interest which has the DNA fragment inserted into it. And that's why we needed to do another copy plate where we can see we have tetracycline this time in the agar. And the plasmid of interest has the tetracycline resistance gene interrupted. So that means the bacteria that have the plasmid with the DNA fragment in should not grow on this plate because that gene has been interrupted. So that means colony A, D and I do have a plasmid, but because they can grow on tetracycline, they must contain the original plasmid because the gene is still intact and therefore they're resistant to tetracycline. Now colonies E and G survived on ampicillin but were killed on tetracycline. So that means colony E and G must contain the plasmid of interest because they are resistant to ampicillin but they're not resistant to tetracycline. And that is how we use those antibiotic resistant marker genes. Now a similar idea is using the fluorescent gene markers, and GFP, which is green fluorescent protein, is a gene that naturally occurs in jellyfish and it can create this protein that fluoresces. So the same idea again, you can have a plasmid which has the gene for GFP and we then have our DNA fragment. We deliberately insert the DNA fragment in the middle of the GFP gene. So that means any bacteria that take up the plasmid with the DNA fragment in will no longer fluoresce. So that's what we're looking for, is when we grow the bacteria, all of these colonies which are not bright green, they must have the gene disrupted by the DNA fragment. So the ones that are not glowing are the bacteria of interest. The ones that are glowing have the original plasmid with the gene still intact, so the DNA fragment didn't successfully get taken up. The last one was enzyme markers, and the enzyme lactase is often used because it can turn a certain substance blue to colourless. So the gene for this enzyme is inserted into the plasmid. Again, it's inserted deliberately in the middle of the gene for that enzyme to disrupt it. All of the bacteria are then grown on the agar plate with this colourless substance. And any colonies that can turn the colourless substance blue, must be the ones that contain the original plasmid without the DNA fragment. However, any that cannot catalyze that reaction of going from colourless to blue, so the colonies remain colourless, must contain the DNA fragment because the enzyme gene was interrupted. So those would be the bacteria that you would then remove and grow large quantities of. Now, alternatively, the DNA fragments can be amplified in vitro, and that means not in a living thing, and that is where PCR is used, which is the polymerase chain reaction. So once you have your DNA fragment, what you would then do is amplify them using an automated machine, and this is the equipment that you would need. The machine is called a thermocycler. You would add the DNA fragments that you isolated that you want to clone. You need to add the enzyme DNA polymerase to catalyze the creation of new DNA polymer chains. You would add primers, which are short single-stranded sequences of DNA to help initiate the DNA replication. And you need lots of DNA nucleotides so that you can create these new DNA polymers. Now, this bit here where it says Taq polymerase, this is referring to the fact that this DNA polymerase is actually taken from bacteria which can survive in extreme temperatures. And that is because this machine is run at very high temperatures. So we need to have a DNA polymerase version which isn't going to denature at high temperatures. So here's our method. The first step is the temperature is increased to 95 degrees C, and what that does is it breaks the hydrogen bonds between the DNA fragment that you added. So we now have single-stranded DNA molecules. The temperature is then decreased to 55 degrees C, and that is to allow these primers to attach, and we call that annealing. The enzyme DNA polymerase can then attach and join any of those complementary DNA nucleotides that align. So it's going to join those adjacent nucleotides, creating the second chain. So we have now created a copy of that DNA fragment of interest. The temperature is increased to 72 degrees at this stage because that is actually the optimum temperature for the Taq DNA polymerase. Now that is one cycle. This machine will be left to cycle over and over, so you're making thousands of copies of that DNA fragment of interest. So the advantages of PCR is it's automated. Once you've added all of those ingredients, you turn the machine on and therefore it will just start working and create the fragments. It's very, very rapid. So you can make 100 billion copies of DNA within hours. And it doesn't require living cells. So it doesn't require bacteria like you needed in the in vivo methods, which makes it quicker and it's less complex. Now, DNA probes are used in lots of the gene technology applications. So what a DNA probe is a short single-stranded piece of DNA, and they are labelled so you can identify where they are. And they're labelled usually either using a radioactive molecule or a molecule that will fluoresce. So that is how we can then visualise where that DNA probe is. And that's what it's used for, locating specific alleles of genes. And this could be used to screen patients to see if they contain a gene of interest linked to a heritable condition. It could be used to identify whether you have a gene that might indicate a particular response you'll have to a drug. So the way that this would be done is a sample of the patient's DNA is removed and we heat it up to make that then single-stranded. You would mix that patient's single-stranded DNA with these single-stranded DNA probes, and if there is a DNA probe which is complementary to the sequence of a patient's DNA, then they will bind together. And these DNA probes can be designed to be the exact complementary sequence to a particular allele, and therefore, if they do align, that means that this patient does have the allele that is known to cause a particular disease. So the final step would be you'd have to visualize this DNA probe, and if you used a radioactive label, you'd use an X-ray machine and that would make that radioactive label light up. If you used a fluorescence, then you'd need to use a UV light. DNA hybridization is when the DNA is heated to separate the double helix and single strands, and this is then mixed with a complementary sequence of single-stranded DNA. So much like we just looked at there in the DNA probes. Once it's cooled, the complementary strands will join together, which is what annealing is. And this method is used with those DNA probes that we just saw in medical diagnosis to see if someone does have a particular allele in their DNA, which is known to cause a particular disease. Now, alternatively, it could be used linked to personalised medicines and genetic counselling. So as well as screening for the presence of alleles, there are other uses also. Now, if you have used it to screen for a particular allele, that will enable the doctors to then select medicines that are known to work better with that particular genotype. Because there are some drugs that will have different effects on you depending on what your genotype is. So some drugs are more or less effective depending on the alleles you have. So it means that number one, the drugs that are selected will be more effective at treating your condition, but also it's more cost effective. You're not taking drugs that aren't going to have any effect, which would therefore have cost just to have drugs that are doing nothing. Genetic counselling is a type of social work where people are given advice based on their results of the top experiments. So if you are screening to see, do you have a particular allele which is known to cause a disease, then genetic counsellors would talk you through, first of all, before you screen, whether you would actually like to screen and what the pros and cons are of finding out if you have that disease-causing allele. And then afterwards, if you do have results that show you have a disease-causing allele, they'll talk you through what your options are now. So it could be they might talk you through options of changing your lifestyle to reduce the likelihood of that disease having any impact, or how you can monitor and check to see if you do have that disease, you detect it earlier, and therefore you're more likely to have successful treatments. So next we go on to this concept of genetic fingerprinting, which is a way to examine DNA. And this uses VNTRs in your DNA. And 95% of human DNA, or sometimes the estimate is actually around 98%, is made up of introns. Introns do not code for amino acids, and they contain VNTRs, variable number tandem repeats. And what this means is you have sequences of DNA just repeated over and over and over. And the probability of two different individuals having the same VNTRs is very, very low. However, the more closely related you are, the more similar your VNTRs are. And therefore, genetic fingerprinting analyzes the VNTRs in your DNA, and it can use this to identify genetic relationships and also variability within a population. Now, genetic fingerprinting is split into these stages. So we'll go through collection, extraction, digestion, separation, hybridization, development, and analysis. Now, collection just means collecting the DNA sample. And if this was a crime scene, well, you have to collect whatever you happen to have. If this is a collection to do a paternity test, you might take the DNA from white blood cells, or it could be DNA from a cheek swab sample, for example. Now, the smallest sample of DNA can be collected, and it could be from blood, it could be from body cells, or hair follicles. Depends if it's a crime scene, what you have available. If the sample of DNA is small, which most of the time it is, then PCR would be used first of all to create a large sample of that DNA. Then we would have to digest this sample, and this is where we use restriction endonuclease enzymes. So they will cut at the recognition sites and we're left with these sticky ends. The DNA samples are then loaded into these holes, which we call the wells, on a gel plate. and the gel is placed in a buffer liquid, and it has an electrical voltage applied to it. Now, DNA is negatively charged because of these negative charges on the phosphate groups of DNA. So the DNA that's been injected into these wells will actually start to travel through the gel towards the positive end of this gel because it's attracted to it because DNA is negative and we've added this positive electrical charge. So this is the stage of how we separate out the DNA fragments that have been injected into the wells. And this is the gel electrophoresis step. So the agar gel creates a resistance for the DNA moving through, and the smaller pieces of DNA can therefore move faster, so they'll move further along the gel. This is how the different lengths of the DNA VNTRs are separated. An alkaline is also added to separate the DNA so it becomes single-stranded instead of being double-stranded, and that is then useful for the next step, hybridization. So we add in DNA probes, which we said are short, single-stranded pieces of DNA that are either radioactively or fluorescently labelled. They have been designed to be complementary in sequence to the VNTRs. So you add these DNA probes, they'll align next to the different VNTRs, and they will then hybridize, meaning join together. The development stage is how we can then visualize where those DNA probes are. Now the agar gel, as it starts to dry out, it does shrink and it cracks. So the VNTRs and the DNA probes have to be transferred to a nylon sheet. And the nylon sheet can then be exposed to X-rays to visualize the DNA probes if they were radioactively labeled, or if they were fluorescently labeled, you would apply a UV light. And what that will then give you is one of these patterns that you might be more familiar with, where it shows you all of the VNTR bands and you can see the positions of them, and then you can identify or do your analysis. So in this case, this was our unknown sample of DNA, and we then had 5 options of organisms that may be our unknown. And you compare the position of the bands, and if they have the same positions, then it is the same DNA. So our unknown, we can see in this example, was the same DNA as organism 3. So that enabled us to identify that the unknown sample must have come from organism 3, because all of the other bands, they don't match up. So this can be used in paternity tests or at a crime scene. It can be used to identify whether the DNA found at the crime scene is matched to a potential suspect, or it could be the victim. It doesn't prove any crime has happened or that the person is responsible. It just proves that a person was present at the crime scene because their DNA was there. So here's another example just showing you these results. This time it's for a paternity test. So we have the mother's gel electrophoresis results. We have the child, and then we've got father 1, 2, 3, and we have to identify which one is the father. Now, any of the bands that didn't come from the mother, which you can see here have been highlighted in orange, must have come from the father instead. Now, in this case, actually, none of these would be the father because none of the bands exactly line up. Number 1 is very, very similar, but they don't actually have that band there. So this paternity test actually shows that none of them were the father. Now, another set of uses are, we've talked about the forensic science, but it could be used for medical diagnosis. So to identify, do you have a particular sequence of DNA that's linked to a genetic illness? Or it could be used to identify how closely related individual animals or plants are before you breed them together, to make sure that you're not going to be doing any inbreeding and therefore potentially passing on harmful recessive alleles. So that is it, topic 8 in a nutshell. I hope you found it helpful. If you have, please give this video a thumbs up and make sure you subscribe so you don't miss out on any of the other whole topic videos.